Yo, tú. Buenos días. Good morning, everybody. Thank you very much for being here in Concepcion, and Happy New Year to everyone. And it's a pleasure for me to give you the welcome for the 12th Ecodim. So it's been more than 20 years since we have been holding this course. And of course, we cannot do it without our wonderful sponsors. You are going to go to a very special place and you will be hopefully very well treated here. And that's thanks to first the Moore Foundation that is giving us support for the last few years. Also from the Agron Institute, they have been together for supporting us for more than 10 years. Yeah, more than that. And also the grad school of Universidad de Concepción, which also supports part of the guests that we have here. Also, we have a partners institution like Universidad Mayor from Nicole. She'll tell you a little bit more about that. Also, they have been working with us in Ecodim, and since last Ecodim 2 years ago, we have also the support from the Winai Foundation. That's where we are going to the scientific station. Originally, this course was held all in Dichato. You will go there tomorrow and visit the place. But since last course, we decided to explore a new environment. A lot of the work that we do during the course is hands-on. You will go in the field, collect samples, ask questions. You will see in the program that Matthias will tell you about it, that this is a very special course because you spend a lot of time in the field, in the lab, doing actual research. And that takes, of course, time, funding, but also the support from people here, that your TAs that are going to be with you, and also from the lectures. I want to especially thank the new lectures. Every course we have new people invited. We have a core set of instructors, but also we have new ones. And the idea of this symposium is that you get to know both a few of the oldies, but also the new lectures, the newcomers. Some of them have gone through Ecodim already, so they are alumni. So that's nice to see that people are returning to Ecodim after many years. And and you will see that one of the, I would say one of the strongest point of this course is to build network. You will see that the people that you meet in the course, some of them are going to become your good friends. Others are going to become good colleagues in the future, and you will see that you are going to go and meet them through your professional career. So this is something that we are very proud of having this course. And again, I want to thank you. We get time to meet during the course, but of course we have some guests here that they are going to be here with us only during the mini symposium. Please talk to them, ask them all the questions. We're going to have a lunch together. If any of your friends or colleagues want to meet our lecturers, please invite them for lunch. There is space, there's room for some guests. So please do so. So again, I want to welcome you in the name of our university, Universidad de Concepción, and also I'm director of the center. It's called the Millennium Institute of Oceanography, which is also giving support and for this course. So I want to give the microphone to Nicole, who is the organizer of this mini symposium, and she has been for the last Ecodims too. So thank you and welcome again.
Good morning, everybody. It's really a pleasure for me. It's an honor also to be here, welcome you to this new mini symposium. Ecodim started in 2000, and since then, I think we have already more than 160 students. This course has gained recognition, international recognition. It's a place where we are gonna try to enhance your understanding of microbial oceanography and marine microbes in several kind of environments. It's a place where we are trying to ignite deep conversation about how we understand marine ecosystem from the microbial point of view. And every year, Ecodim held this wonderful symposium. At the beginning, the original name was Mini Symposium, but I think now should be named Symposium. It's a big symposium, really. So, but it's really important because this is one we try to connect to the overall sea. It's not just because here are lectures that are going to train you, it's also because we want to know all the community, the scientific community about Ecodim, about the importance of microorganisms in marine environment. In this particular course with this symposium about microbial life and symbiosis are trying to understand that one of the main or one of the more important microbial type of life is at the end the one that is in association. And I will try to stretch you that you think about this in other topics of scientific life. Scientific life could not be done alone, and I think microbial life could also not be done alone. So I really welcome you. I thank you for being here, for the people that is now in this aula, but also for those that are by streaming following us. Normally we see after this the point around the globe, and we see that all the students that are spread around different places are following every 2 years this important activity. I wish a productive, a very stimulating discussion today about different topics. We will have Chilean lecturers or speakers, international speakers from different fields, from different university. We try also to be as gender equal as we can, as institutional diverse as we can. So please enjoy and give at the most of the advantage of this opportunity that you have today. So welcome and thank you for the speakers, for those that are following us, and enjoy the day. Thank you.
Thank you, Nicole. And now, just a few words from our Deputy Dean of the faculty where we are holding this symposium. Victor?
Good morning to all. I will do my best. My English is not so good, but it's to give you a warmest welcoming to the university, to our faculty, and of course to City of Concepcion, received with open arms. And sorry, todo de nuevo. In this summer school for the university. Now it's opening by the rector in the forum at this time. All the activities for this month, academic activities, artistic activities, for all the people who visit us in the university. I would like to say thanks to professors, to colleagues who is participating in this course, Dr. Orlando, Julieta Orlando, Dr. Matias Castro, Dr. Nicole King from University of California. Thank you for your visit. Dr. Adriana Lopez dos Santos da Universidade de Oslo, né? E a doutora Leslie Dial. Thank you for your visit and for to stay here with us in this Ecodim, and the version is 12 version. Very good. Thank you.
Okay. During Ecodim, during these years, there are several like traditions that we try to, things that we develop every 2 years in every Ecodim, so you will be like printed with different kind of stories and things. One of the tradition is that in this symposium, every speaker is presented by one of the students. That approach the student to know the curricula, the life, trying to see how can be best presented or introduced. And in this case, Joanna from Argentina will present Doctora Julieta Orlando, that is also from Argentina, so we are trying to make like a kind of match, international match. So thank you very much, Joanna.
Well, welcome everybody. It's an honor for me to introduce our first special guest, Dr. Julieta Orlando. Dr. Orlando is a professor at the University of Chile, where she also serves as the Deputy Director of the Millennium Institute Biodiversity of Antarctic and Subantarctic Ecosystems. In addition, she is Vice President of the Chilean Society of Microbiology. She leads a team exploring diverse microbiomes. Using metabarcoding, metagenomics, and functionality predictions, they unravel selectivity, functional roles, and phylogeographic patterns. Their research offers insight into interspecific associations, microorganisms' roles in diverse environments, and bridge the gap between macrobial and microbial biogeography. Today, she will share her insights in a talk, Microbes at the End of the World, from a community to an intra-population perspective. Please join me in giving a warm welcome to Dr. Julieta Orlando.
Okay. Thank you very much for the presentation, Joanna, and thank you. It's okay. For the invitation to this interesting big symposium. Today, I will talk effectively about microbiomes at the end of the world, from a community to an intra-population perspective, and as Joanna mentioned, I'm representing Universidad de Chile, but also the Millennium Institute Base, and the SOMICH is the Chilean Society of Microbiology. I will use this point. Okay. In this opportunity, I want to show you a general presentation of one of these institutions, the Millennium Institute Base. Next, I will present some examples of our research related to microbiomes of marine animals. This is a recent topic in my research line, so this is my first time in Ecodim, because I'm more terrestrial microbial ecologist. And then, of course, I want to show you an overview about some studies on terrestrial environments, but connected with the marine sites. And to finish, I will share with you 2 outreach activities that we perform in the Institute. The Millennium Institute BASE, the acronym of Biodiversity of Antarctic and Subantarctic Ecosystems, is funded by the Millennium Initiative Program from the Chilean Research and Development Agency for 10 years since December 2021. When we submitted our proposal for the Institute during the summer of this year, 2021, for the first time, the International Committees on Biodiversity, IPBEs, and Climate Change, IPCC, published this report together, warning about the necessity and importance of putting the nature at the center of decision-making. So, across these 8 research lines, we propose to investigate the past, present, and future of Antarctic and subantarctic biodiversity, contributing this way with knowledge to support decision-making in 2 important areas, conservation and governance. The host institutions of the proposal are Universidad de Chile, Pontificia Universidad Católica de Chile, Universidad de Concepción, Universidad Austral de Chile, Universidad de Magallanes, and Instituto Antártico Chileno. In addition, during these 3 years, we established collaboration with several strategic partners, including other centers of excellence with complementary objectives such as IEB, CHIC, IDEAL, and CRG. This associative research connects researchers from different disciplines and institutions from Santiago to Puerto Williams. Puerto Williams is the last city in Navarino Island in Chile. We have more than 30 researchers, including the main associate, senior, and young researchers, which include postdoctoral and adscript researchers. And in addition, we have more than 100 students who are developing their thesis in topics related to the Institute. And as you can see here in the clip, they are also our ambassadors in many of the outreach activities that we perform across the country. Last but not least, of course, I must highlight the vital support that we have through professionals in management, administration, communication, of course, scientific activities. A significant number of researchers from the Institute, including many young researchers, students also, have been fortunate to participate in multiple Antarctic campaigns, covering sampling points not only in the Antarctic Peninsula and the surrounding islands, but also throughout different parts of the coast and also the South Pole in the Antarctic territory. And moreover, we had 2 Arctic campaigns, in one of which I had the opportunity to travel to the geographic North Pole with the different biodiversities in both poles being the objective of comparison in this research. These campaigns are possible thanks to the collaboration of the Chilean Antarctic Institute, but also from a French tourism agency that is named Ponant. During our field activities, we collect samples and data of different components of this Antarctic biodiversity from the, of course, more emblematic organisms such as penguins and other birds, like here petrel, and also cetaceans and different fishes here between the rocks. And of course, we also study key components of the ecosystems with emphasis, for example, in the krill, that is the key crustacean in the food web of the Southern Ocean. But we also include organisms less known for different reasons, because, for example, they are difficult to access. Okay, difficult to access, or they are small in size, and among other reasons. In this photograph, you can see a sea urchin, a limpet, and some sea snails as examples of these organisms. The diversity of marine animals in Antarctic is impressive, but in the terrestrial environment, the diversity is more restricted. We can highlight some freshwater invertebrates. Here you can see Branchynecta, and in red, the red points there are Bokella, a copepod, a little copepod. They live in freshwater systems in the terrestrial environments. You can see here the only winged insect native to the continent, that is Paroclus steineni. And of course, there are also different components of the flora. There are not trees in Antarctica, but there are two vascular plants, the Schampsia antarctica and Colobanthus, and they are accompanied by different mosses and a lot of different species of lichens. As you can see, the Antarctic biodiversity is much richer than many think, including, of course, a great diversity of organisms that are not visible to the naked eye. And although microorganisms are not only the study models that we have, we are exploring the microbiomes of diverse environments. As you can see, soil samples associated with different animals or plants, mosses, lichens, water from the sea environment or fresh water in the terrestrial one. And now I will share with you, because the topic of the symposium, some of these studies along with the work in progress, beginning with the microbiome of marine animals. At the beginning, in this line, our first study was using as a model Abatus agassizi, this sea urchin that you can see here in this picture. First, we compared the microbiota associated with the gut tissue, the gut content, and the sediment where these animals live, and we found that the microbial diversity in the gut tissue is less diverse and different from the other two microenvironments. These were sampled in two different localities, located next one of each. And we also performed this network analysis, detecting key taxa in the triangles that have potential critical functions for the host. For example, spirochetes able to fix nitrogen, which could provide this nutrient in a context of low nitrogen food. And another key taxa are related with sulfate reducers that could contribute in degrading recalcitrant organic compounds present in the sediment. Then, analyzing the key OTUs detected in the communities in the last work, we perform microdiversity analysis to study the intra-population phylogeography of bacterial taxa across the Southern Ocean, the ocean that is surrounding the Antarctic continent. In this study, we found three main patterns. The first one, a genetic homogeneity within the Patagonia province, as you can see here in these squares, where the blue arrows are strong connectivity inside of the Patagonia province. The second pattern was a barrier to dispersal between Patagonia and Antarctica. There are no connections among these two provinces because the Antarctic polar front. And third, we detect connectivity between Kerguelen Island, this island here in green, with maritime Antarctica and with Patagonia for different OTUs due to the Antarctic circumpolar current. In a more recent work, we examine how the habitat specificity influences the bacteria biogeographic patterns. And as we previously determined these three environments related with the sea arcing, present or exhibits a different specificity. And in this sense, the gut content is the more specific habitat in comparison with the gut content and the external sediment. The results show that the bacterial communities exhibit a stronger geographic and environmental influence and divergence in less specific habitats, such as sediment in comparison with the gut tissue. As you can see here, for example, in the comparison of the distance decay relationships slopes that have this pattern. These findings so highlight the importance of considering this habitat specificity to understand the microbial distribution at one scale that covers different parts of the ocean. Now, using a different model, in this case limpets of the genus Nacella, we investigated the ecological filtering and phylogeographic structuring of some bacterial taxa in two closely related species, Nacella deurata, located in the West Antarctic Peninsula, and Nacella consina, located in the Falkland Malvinas Islands. Our findings reveal specific bacterial communities in radulas and intestines, which are different, significantly different from the ones in the surrounding environment, represented in this case by the rocks used by these animals as substrates. A specific OTU related with Cycriliobacter exhibit a strong phylogeographic structure in the intestine, but not in radula and rocks, forming two distinct clusters specific to each Nacella species. This suggests potential co-diversification between the limpets and this member of their microbiota. These results show highlights the importance of Nacella limpets as a promising model for studying the host microbiome interactions and evolutionary relationships. And this was actually, I think, shared by the reviewers of a proposal based on this information, as Dr. Guillaume Schwab was recently awarded with a funded initiation project on this topic. Now, using arpahifer, this fish, as a model, we investigated the cophylogeny and phylosymbiosis between fish species and their good microbiomes, also across the Southern Ocean, and we identify a low diversity core microbiome dominated by Alivibrio. And this suggests a potential mutualistic relationship and a possible selection against these bacteria. In addition, we determine phylogenetic congruence between marine fish and its microbiome, which highlights an intimate association between hosts and some members of these microbial symbionts. This could be performing ecological relevant roles, but we need to deep in the genomes of these organisms to confirm this suggestion. These results underscore the importance of evolutionary history in shaping the gut microbiome in marine fish. In collaboration with students of different programs and various researchers of the Institute here in brackets, we already have expanding the study models beyond sea archings, limpets, and fishes, and we are now studying marine sponges. So maybe we could collaborate in the future with Nicole and her group. And in each, not just the marine organisms, but also we are beginning studying the microbiome of the terrestrial animals, such as insects and freshwater invertebrates. In each case, we search for potential contributions of the microbiome to the host physiology. We evaluate these patterns of coevolution and philosymbiosis between hosts and their microbiome. And we are assessing the impact now of multiple stressors such as temperature, pH, salinity on the diversity, structure, and function of this microbiota of the different hosts. In addition, we are also studying, it's not working, okay. We are also studying biogeographic patterns of bacteria in high-latitude lakes, terrestrial ecosystems, considering different levels of the ecological organization, with emphasis in the methane cycle. And we are evaluating the influence of the Antarctic polar front on the genetic and functional diversity of microorganisms in seawater of the Southern Ocean, specifically in the Drake Passage. Okay. Now, I want to tell you briefly about the research on the microbiome of penguin soils. Soil, not poop, is the sample. And talk about soil samples at this symposium, a big symposium, may seem out of context, but penguins, as marine animals that develop part of their life cycle on terrestrial environments, are good links in the transition between the marine and the terrestrial ecosystems. So, a time ago, in collaboration with Dr. Nicole Treffold, we studied through 16 metabarcoding pyrosequencing in that time, the bacterial communities in soils from the Cape Shirreff, this small point in this big island, Livingstone Island, in the South Shetland Islands, impacted by different marine animals, birds and pinnipeds, and we found that the soils impacted by the penguins were the most different from the control, and that nitrite was the most influential edaphic factor on these patterns. So, next, with interest in depth about the bacterial diversity in these soils, we use metagenomics, including an additional point in the South Shetland Island, the Fields Peninsula in King George Island, and we assembled in that opportunity 100 metagenome assembled genomes, more known as MAGs, and searched genes potentially related with the emission of nitrous oxide, that is a potent greenhouse gas, finding that the relative abundance of genes coding enzymes related to the nitrification was at least double in soils impacted by birds than in soils without animal influence. It is pending to measure if the nitrous oxide is actually releasing from these soils, because this could be an important source of this greenhouse gas. Due to the importance of penguins in impacting the soil microbial diversity, we sampling soils impacted by these birds along the Antarctica Peninsula, having now more than 80 metagenome sequencing from different islands and part of the peninsula, considering the 3 different species of penguins in Antarctica of the genus Pygocelis. And we are assembling the MAGs. We have more than 1,000 MAGs already assembled. And in those, we are searching for differences among localities and the penguin species that we sample. And preliminary results indicate that the penguin species seems to be the most influential factor on the microbial diversity in these soils, independent of the location of the samples. In addition, we are predicting microbial functions related to greenhouse emissions and nutrient cycling, but also microorganisms able to, or potentially able to, biodegrade different pollutants among fuels and microplastics. And this last topic is the main objective of a funded project awarded last year, 2024, in collaboration with Dr. Lea Cabrol. In brief, this project is focused in exploring the possibility of penguin-impacted soils as a potential reservoir of microplastic degrading microorganisms in Antarctica. The rationale of our proposal is that plastics degrade into microplastics, which are ingested by marine animals, including penguins, which could transfer these contaminants or the microorganisms capable of degradate to the terrestrial environment and promote microorganisms capable of metabolize these microplastics. We hope the results of this ongoing project will contribute to design remediation strategies of Antarctic soils in the future. And I don't want to end without showing you two of the initiatives we carry out about outreach. The first one is the Antarctic Metaverse, that is a video game that allows players to explore 7 Antarctic ecosystems of different colors, because we have the green Antarctica, the blue Antarctica, also, of course, the white Antarctica, among others. In that, the players can collect samples of native species and identify pollution sources to help protect biodiversity. This project was developed by the members of the XR Labs at Universidad de Chile with the scientific guidance from researchers of the Instituto Milenio Base. And the other project, ARA, because the Spanish meaning of Antarctica in augmented reality, introduced 6 Antarctic species through cards that can be scanned with smartphones using the Instagram filters, and you can download these cards from the website of the Institute. And when you scan, this brings to life through augmented reality the animals that are represented in the cards, and the back have information about the species with emphasis in their ecological importance. Both projects, Metaverse and ARA, have achieved great success in different audiences through activities in schools, science fairs, and different media platforms. So finally, I invite you to follow us on our social media, Milenio Base, and thank you for your attention.
Thank you very much, Dr. Orlando. It was a beautiful talk. You covered different topics, and I'm really glad to hear about Antarctic research and microorganisms in Antarctica. We have time for a couple of questions. Come on, don't be shy.
So early.
Jake, please.
First, I really enjoyed your presentation, and particularly the art illustrations were really fantastic. I wanted to ask about the urchin microbiome that you saw sulfate-reducing bacteria in there. I was wondering if you also see sulfur-oxidizing bacteria that may detoxify the sulfide that's produced by the sulfate-reducing bacteria.
Yeah, we have no more information than the general diversity of this microbiota, but we are now working in obtain the transcriptome, because we predict the possibility of these activities that help the physiology of the sea urchin that is feeding on sediment, and the sediment is low in different nutrients, but with the prediction we have not approved that the microbiome is helping the animal, but with the transcriptome we are going a next step in improve these activities, but it's really difficult to work with RNA in environment samples that are preserved normally in ethanol, because the animals are preserving this solution, and the RNA is not working in this sample, so we need to improve the sampling in campaigns that are difficult, for example, transport liquid nitrogen, because in the flights in Antarctica or the long trips in the ships are logistically challenged, but we are working now in known in more detail the microorganisms in the sea archin and different animals in Antarctica.
Great, thank you.
Thank you. Another question? I have one.
No sponges, please.
No, I have a question about the philosymbiosis that you show. I think it's a wonderful topic, but the philosymbiosis signal that you detected were in all the microbiome or was specific to some taxonomic groups?
Yeah, it's important the difference because there are a gradient of relationships among the phylogeny of the host, different hosts, and you can have a congruence, for example, of the whole community when you use a clustering method, for example. But when you want to study the symbiosis in particular, you must go intrapopulation, detecting first OTUs, for example, and then analyzing this at the microdiversity level. So the association of the phylogeny of this species or intra-species level of the organism, in this case with Sarpahifer, was a OTU of the Alivibrio genus, and there you can have a proof of possible philosymbiosis, but you also need to discard different components that affect the diversity of this symbiont, because there are deterministic and random processes affecting the diversity. So you need a lot of samples from different species that have a good phylogeny. It's not the case of Arpahifer. This was difficult because maybe the microbiome could be used as an additional, for example, information in defining the species levels in the microorganisms hosts. So it's difficult to prove, but are steps that you can analyze to predict a congruence of the evolutionary history.
Perfect. Thank you very much, Juliet. The last question? No? Okay.
In the coffee.
In the coffee, absolutely. Thank you very much, Julieta.
Thank you.
Our next speaker. Wait. Okay. Yeah. Our next speaker will be introduced by Claudia Talia from Cuba. So thank you very much for introducing our wonderful new speaker.
Good morning. Dear colleagues and attendants, it is an honor to introduce our next expounder, Dr. Matias Castro, who has a solid academic background and outstanding science record. He ascended in the degree in molecular biotechnology engineer of the University of Chile and continued this training in the molecular microbiology by completing a PhD in microbiology and institutes in both Chile and South Africa. Dr. Castro specialized in the molecular mechanics of bacterial colonizations and biofilm, including signal transductions, cell-cell communications, motility, and the production of exopolysaccharides and membrane besides. He has collaborated with numerous national and international institutes and research groups. A few years ago, as a researcher at the Millions Institute of Oceanography, Dr. Castro initiated a new line of research focused on the characteristics variances in the deep ocean, which is the sum of today's lessons. His dedicated and passions as a microbiologist at evidence, and we are as excited and grateful for your presence in this symposium. Thank you.
Hello, everybody. Thanks, Claudia, for the presentation. I'm going to present today a very exploratory work performed by the Millennium Institute of Oceanography in collaboration with the Ohio State University. It is about viruses in the ocean, but in the particularly in the deepest point of the ocean that we call trenches. So we know that the viruses play important roles in the ocean. Viral particles are the most abundant biological entities in the ocean. 1 to 10 millions in a single drop of seawater, which is very impressive. And viruses infect macroorganisms, but also microorganisms. Importantly, controlling their population. The microorganisms, on the other hand, are the most abundant cellular organism in the ocean, where they perform important ecological processes and also biogeochemical cycles. And well, the viruses control the population of microorganisms, basically killing them. But these also produce and release dissolved organic matter, and also produce sticky aggregates of organic matter that sinks to the depth. Since bacteria are the most abundant microorganism in the ocean, the viruses that infect bacteria that we call bacteriophage are the most abundant and probably the most important virus in the ocean. Bacteriophage, as well the other kind of viruses, are not so easy to classify, probably because all of them are kind of chimera. But we can see one way to classify them is based on their genome and how this genome is covered or protected by protein or lipid envelope. So we have a lot of mixes of that. We have double-strand RNA viruses, single-strand RNA viruses, single-strand DNA viruses, and probably the most known, studied, and maybe the most important, the double-strand DNA viruses, which can be non-tailed, but also tailed with these beautiful shapes, with a head, with a tail, and with these like spider legs. well, the infection of the viruses imply or need the delivery of the genetic material inside the host, and these promote the horizontal genes transfer by a lot, or at least by 3 different mechanisms. Uh, in the lytic cycle of the virus, in the replication step, where the viral particle are assembling, sometimes the the viral particle packed some piece of the DNA of the host, and when this viral particle infect a second host, this DNA from the previous host can recombine with the host chromosome. So the second host can gain or acquire new functions. That is very powerful. On the other hand, during the lysogenic cycle, in which the entire DNA of the phage get integrated in the host chromosome, well, the phage can resume the lytic cycle, but before they need to be excised, and sometimes the phage machinery make a mistake and could beyond the DNA of the phage, taking some pieces of the host DNA. Then it can recombine, and again the second host can gain some function. And in a third pretty big mistake, the phage machinery sometimes start to pack the DNA of the virus without excision. So of course the viral particle pack the DNA of the virus, but along with a few kilobases of the genome of the host. Moreover, the phage can live in symbiosis with the host. The host provide environment that allow the expression of the genes of the phage, and if the host is lucky, that some of the phage genes can be useful to face some particular environment for the host. That is very important. This is very powerful. We can think about the cholera disease. We all know that the cholera disease is the cause is our pathogenic strain of Vibrio cholerae, and the difference between the pathogenic and non-pathogenic strain of Vibrio cholerae is a phage, which encodes the cholera toxin. So this is very powerful, and the cell that is infected by the virus is never like before. Of course, it's not the virus, but it's not the cell. It's a new entity, a completely new entity with new capabilities, which depends of the viral particle that is infecting the host, depends of course on the host itself, and also depends on the environment that will select or not a particular virus cell. The virus cell concept is very interesting because correspond to the life phase of the phage, whereas the the particle, the viral particle are like the spores or the seeds of the plant. Some of genes that the phage are sharing with the host, some of them have been characterized. They are involved in photosynthesis, for example, or in the central metabolism. So quickly, the authors that have been studying this call these genes auxiliary metabolic genes. But we will see that not all the genes that are spreading by phages are directly involved in the metabolism. So the characterization of virus could be a good step to start to characterize new environment, because you can ask who are the virus, but also who are the host, and also, and importantly, which kind of genes they are spreading that could be helping to the virus cell to thrive in some extreme environment. So one of the less understood habitats on Earth are the trenches, oceanic trenches. They correspond to the deepest point of the ocean. Of course, this is a very extreme environment. There is not sunlight. The temperature is very low, and the pressure that the hydrostatic pressure that is reached in the bottom of the trench are huge. As a Chilean and as a research of the Millennium Institute of Oceanography, I'm very lucky because we have access to the Atacama Trench or the Peru-Chile Trench. This trench, well, is located from the south of Peru to the north of Chile. The depth in the deepest point reach 8,000 meters, and the pressure there is around 800 atmosphere. Unlike the most of the other trenches, Atacama Trench has an oxygen minimal zone in the surface, and also is influenced by a current, humble current, which allow high primary productivity. The result of this is a lot of particular material, organic material, and here there is a paradox, or at least difficult decision for the microbes, because could be very nice, good to attach to this particle to gain nutrients, energy, but also they are going to sink and need, and if they are going to sink, they need to deal with this tremendous pressure. So we decide to start to identify and characterize the viruses present in the Atacama Trench, and for that we collect 13 different samples in different depth with different instrumentation, and the water was filtered by different pore size, and we have two kind of sample. The particular material we can call large particles between 20 and 3 micrometers, and the small particle between 3 and 0.2 microns. We perform DNA extraction, sequencing, we assemble metagenomes, and we use state-of-the-art bioinformatics tools to, well, predict, first of all, the viruses, well, check the quality, determination of the viral population. We try to predict the host And finally, we annotate and we try to identify the auxiliary genes that these viruses can be spreading. So we identify near to 19,000 viral sequences in the Atacama Trench. This is a similarity network. Each point represents a virus, and the color represent the depth. The more light the color is in the surface, more dark is in the deep. And the pattern here is not very clear. Here on the right we have, well, the depth here and the number of viruses in the x-axis, and we can see that the most of viruses we found are in the surface, and then they decrease, but then again increase near to the bottom of the trench. If we split this in the small and large particles, in large particles we see something like before. A lot of viruses here, then decreasing, and then rise again. But in the small particles, we have just a few in the surface, and then they increase near to the bottom. So here are at least two possibilities. One is there are more large particles at the surface, which gradually consume on the way down to the bottom, where the smaller particles are more abundant. That is one. The second one is that the microorganisms and the phages may shift the attachment preference from large to small particles, for example, to reduce their sinking rate. Yeah, if this is true, this suggests that regulating the transition between free-living and particle-attached lifestyle, like a biofilms, could be a good adaptation that enabled microbial survival at different ocean depths. Okay, but who are the viruses here? We identify 11 classes of viruses, but near to the 90% of them belong to the Caudoviricetes class. Well, these classes are infecting eukaryotic hosts. We have Megaviricetes that infect algae or protists. We have animal hosts, arthropods that we know that there are in the Atacama Trench. We have virophage-like viruses. Those are very interesting because need a second infection of a helper virus to perform their cycles. But as I said before, the most abundant viruses are infecting prokaryotic hosts. We have here just a few of viruses that are infecting archaea, and most of the viruses are infecting bacteria. All right, so these classes, we can see the distribution by depth of these classes, and we can see here that the viruses are in the whole water column. We have some viruses that apparently are confined in just a particular point of the water columns, but probably this is because we don't have enough data. So it's not so clear for me this kind of distribution. Again, if we split this by particular size, we can see that within one class, we have the in the large particles, the viruses are concentrated in the surface, but in the small particles, they are concentrated in graded depth. We can see here in caudoviricetes, but also in megaviricetes, even in these maveviricetes. Here that pattern is not so clear, but could be again because we don't have enough data. Okay, which kind of host we have? We can determine by the IFOP only the 10% of the host of the virus. The viruses, most of them are bacteria, but also we identify some archaea. The archaeal hosts are plotted here. We determine or identify 13 classes. The most of them belong to nanoarchaea that are very interesting organisms. They are very little. They have little genomes, so they can't survive alone. They grow attached to another kind of archaea and share metabolites between them. Also, we have a lot of archaea involved in methane processes and also involving in nitrogen cycles. We have a lot of archaeal hosts. I'm showing here just the top 10%. Most of these hosts are Pelagibacter that we know that is very abundant in the ocean. We have Vibrio. We talk about the Vibrio cholerae, but Vibrio has species that are important symbiont of squids, for example. They are luminescent microorganisms. And we have Alteromonas, Pseudoalteromonas, which are known heterotrophic bacteria, but we also have here Synechococcus, for example, a photosynthetic bacteria, and here we have Thioglobus. This is a sulfur globe. This bacteria is a sulfur oxidizing bacteria. And here we have Alkalinoborax, which is very interesting because it rises in the spot of oil accident in the ocean. All right, so what kind of auxiliary genes are spreading these viruses? We identified something like 10,000 genes, but I am going to focus in just a few for this presentation. One of the most interesting enzymes for me are these 2-oxaglutarate iron oxygenases, which can perform a lot of reactions, are very versatile enzymes. But one reaction that could be very important here is the desaturation of lipids. So this could be a good adaptation to control the fluidity of the membrane. That could be useful to deal with the high pressure in the trench. The other one, sorry. Well, these enzymes are very interesting, not just for that, because they can also perform hydroxylation of proteins, particularly ribosomal proteins, so they can control the translation of proteins. In that way, they can control a more um, general physiology of the cell, and also can form rings like that are need for the production, for example, to antibiotics. And also this is important because this enzyme is integrating three important things. One is the oxygen, the other is the iron, and the other one is the 2-oxaglutarate, that is an important metabolite in the cell. The other auxiliary genes are related, here in green, are related with the sugar metabolism, but also a lot of them are involved in the production of exopolysaccharide and lipopolysaccharide. So again, we have here enzymes that can modify the envelope of the cell, and maybe that is a good thing to deal with the pressure. Finally, I bring three examples of the viruses that we identify. Here is a beautiful T4-like bacteriophage. We know that it's a T4-like because the structural proteins here in purple. Here in pink we have the recombinase, so it can be integrated in the host chromosome. And here we have the enzymes that we talked in the previous slide. Here are the 2-oxaglutarate iron oxidases in red, and in green, light green, we have the sugar metabolism or lipopolysaccharide and exopolysaccharide related enzyme. Very interestingly, here we have some one gene that apparently encode a short version of a mechanosensitive channel, which is located in the membrane and transduce the pressure signal from outside to the inside to the cell. This is the second one. Also is a T4-like, and it apparently is involved in spread genes involved in the transition between biophil and planktonic lifestyle of the bacteria. Here we have again the recombinase, but the important thing here is this gene, which encodes D1-elite cyclase. This enzyme here produces a second messenger called CDGMP, and this second messenger has in the cell a lot of CDGMP binding receptor or effectors related with this transition. The CDGMP can bind a pill-Z domain and repress the motility, specifically the flagella motility, but also can activate the production of exopolysaccharide. So when this concentration of the concentration of this CDGMP is high, the cell decides to attach, but when the level of CDGMP are low, the cell prefers to swim, go away. Okay, so interestingly, this enzyme, the enzyme encoded in this gene, have the catalytic domain that produce the second messenger, but is coupled to a ham domain that is known for get stuck in the membrane. So there is some signals that are sensing in the membrane that the cell can transduce in different level of this second messenger and then control this transition. Finally, we have here a lambda-like bacteriophage. This is beautiful because apparently it is spreading a couple of genes, LuxI and LuxR, that are involved in cell-cell communication, a quorum sensing system. We know that the quorum sensing system involves the production of autoinducers that diffuse from the inside to the outside of the cell, and when the cell population is very high, of course the autoinducer levels are very high, and they then can get inside again to the cells and bind LuxR, which is called here, and when LuxR is when the inducer is binding by LuxR, this is a transcriptional regulator that can activate the transcription of genes that, for example, in Vibrio fishery are involved in luminescence, but in a lot of bacteria are related with the biofilm formation. This is some conclusions. There is a larger number of diversity of viruses in the water column of Atacama Trench. The viral population seems to be more related to the size of cesium particles than to the depth of the water column. And the viral host are bacteria and archaea with metabolic attributes important for the functioning of virus biochemical cycles. And very important, many of these auxiliary genes that are being spreading by the viruses target modification in the cell envelope and also regulate the ability to attach to or detach from cesium particles, including signal transduction mechanisms from membrane stimuli and cell-cell communication, indicating that importance of this process in the Atacama Trench. So thank you to the institution that make this work possible, and thank you for your attention.
Thank you very much, Matthias. We have questions, please.
Sorry if I missed this, but do you see any difference in AT versus GC bias in viruses that are?
Sorry, I'm asking. Is this working? Okay. I'm asking whether you see any difference in AT versus GC in the viruses that at different depths, because I'm imagining that getting the strands to separate under very high pressure might be facilitated. I mean, I'm just hand waving here, but
yeah. Sorry, are you asking for the GC
double-stranded? The double-stranded.
Yeah, but the GC content.
Yes.
I have a slide of that, but not
okay. That's right.
We can see it later. But the pattern is not so clear. Mm-hmm. Yeah.
And do you think that's because they're cycling so much? Okay.
Yeah, I think they are sinking. I think that they are spreading of at least now. We are still with the analysis, but apparently there is no big difference about it.
Thanks, Matthias, for the presentation. It was super interesting. My question is related also to the depths, because you brought this hypothesis, and sorry if I lost, that they are switching between being attached to more planktonic And so I'm assuming it's the same, there is very similar population between the surface and when it's increasing into the depth, or it's completely different population of viruses?
We play a lot with that kind of data. The 19,000 viruses can be dereplicated in like 13,000 population, and you can map that population through the water column, and you have some populations that are only in the surface, and others that are only in the bottom, but you have also a population, viral population, that are in the whole column. So again, the pattern of the depth is not very clear. So I don't know really.
Okay. And just other quick question, when you showed the host and you showed the number of viruses also by depth, I was surprised that for Synechococcus you have more at the...
Yeah, because they are thinking.
Yeah, exactly. But then again, this is the same virus that you're gonna find in the surface, or because it could be just attached to the particles that are sinking and not actually.
Sure. I don't remember exactly that.
Okay.
Yeah, but I can say that well, we can map that in the depth map that we have. Yeah. I can answer that maybe tomorrow.
Yeah, no, we can definitely discuss. Yeah.
Okay.
Thank you.
Thank you very much. Some other questions? Yeah. I have a question. Just if you think about, Matthias, I was surprised about the idea of identifying the host and this link between some archaea and some bacteria and the size of the particle. Do you think that you are identifying the bacteria that is the host based on the size of the particle? I am thinking, for example, in this beautiful paper about common goods in Vibrio, for example, and the size of the particle, in which you can connect with the size of the particle, the depth of the sinking, and the kind of bacteria that is being the host of the virus. Do you think you can also have like kind of connection between them?
Yeah, I think there is a connection, but I have no clear idea yet about that. The prediction of the host was made with a bioinformatic tool called IFOP, that is the best now. But as I say before, with that tool, we just predict the host just for the 10% of our virus. It's very little. So now we are working in trying to expand the database of IFOP with our metagenomes. So yeah, we will improve that soon.
And sorry, and do you have also the community composition at each of the microbes at each of the depth to see if the host is also in your...
Yes, we have the data, but we have to process that.
Okay.
Yeah.
Okay. Thank you very much.
Thank you.
Other questions? Jake? No? Okay. If you
Thank you.
are okay, thank you very much, Matthias. With this session, with these speakers, we close this part of the morning. I thank you to our two speakers, and we are having now a coffee break, and we come back in 30 minutes or something, perhaps a little bit more, because we are on time. So thank you. first speaker is Dr. Nicole King, that will be presented by Matías Castro from Chile, Región del Biobío, Chile.
Hello, everyone. It is my pleasure to introduce Dr. Nicole King, a PhD from Harvard University. She is a professor in the Department of Cell and Molecular Biology at the University of California, Berkeley, in the United States. She also is a researcher at the Howard Hughes Medical Institute. In 2000, she initiated her line of research, which aimed to address one of the fundamental questions in the history of life: the origin and evolution of animals. To this, she uses conoflagellates such as Salpingoeca rosetta as a model organism. These protozoan relatives of animals express members of various protein families essential for cell signaling and adhesion in animals, including receptor tyrosine kinases, caderins, and C-type lectins. The finding of signaling and adhesion gene homologs in conoflagellates raises questions about how these genes function in the unicellular common ancestor of conoflagellates in animals, and also what role they play in the origin of multicellularity. In this presentation, Dr. Nicole King will tell us about the secret life of conoflagellates. Please welcome, Doctor.
Thank you for that kind introduction. And I also want to say it's just such a pleasure to be here. I look forward to getting to know all of you and talking science with you. So I'm looking forward to a really wonderful experience. I'd like to encourage students to ask the first question. So when the talk is over, hopefully a student will be ready. And if you feel uncomfortable asking in English, it's fine with me if you ask in Spanish and maybe someone can translate. But if we can break the ice on that, I think you'll get a lot more out of these talks if you feel comfortable asking questions. Just know they're all good questions. Okay, so the real title of my talk is The Secret Sex Lives of Cuernos, but I didn't want to put that in text. So what I want to talk about is, or what I want to show you with this talk today is an example of where you can really focus on one problem in great depth and detail and learn things that actually have broader importance. And so, you know, there are lots of different ways to do science. So this is a different way to do science where you're kind of narrowly focused on one question and you see what you can learn about it. Okay, so I want to start by giving you some context and just lay the foundation, saying that many of us are interested in diversity. And for myself, I'm very interested in animal diversity. And it's incredible to me how very different all these different animals are. They have very different shapes and sizes. They live in their environment in very different ways. Some of them can fly, some of them can swim. You know, these sponges are actually eating by filter feeding. There's many, many different ways to be an animal. And yet, all animals are what we call monophyletic. They all share a common ancestor. And something else they all share is that they all start at the same place. Every animal, including each of you, starts as a single cell. A single zygote, the result of fertilization of an egg. And then, through rounds of cell division, shown here as these cleavage stages. Each of these animals developed through essentially cell division and cell differentiation, okay? And so this is what happens. This is the unfolding of development in each animal's life. But we can also think about this transition from being single-celled to multicellular in evolutionary time. And so here what I've done is take some of these animals now and put them on a phylogeny to show that they all share a single common ancestor, and then to tell you that what really has captivated me in my career and in my research is to understand this transition from single cell to multicellular over evolutionary timescales, and try to understand how did multicellularity evolve on the lineage that led to animals? How did mechanisms of cell differentiation evolve? And how did mechanisms for allowing cells to coordinate their function first happen? Okay, so this is the talk that I would often give you. We would focus on these kinds of questions. But today I'm going to talk about something different. I'm going to talk about the birds and the bees. Hopefully that's an international analogy. And what we're particularly going to focus on is this question of how do we get this transition from being asexual to sexual, and also think about how we get these differentiated gametes, okay? So what I'm going to do in my talk is first introduce you to the organisms that we study in my laboratory, the choanoflagellates. I'm then going to spend most of my time talking about what we've learned about how both the environment and the genetics of the choanoflagellate intersect to regulate mating. And then I'm going to close with some thoughts about how this study might inform us when we think about animal evolution more broadly. And please, if at any point it's not clear, just interrupt me. I'm happy to be interrupted. Okay, so this is a expanded tree of life where I really want to focus on choanoflagellates and animals. And I want to make a few points here. So here now, again, are some representative animals, making the point that they are monophyletic. And if we compare animals in this phylogenetic framework, we can find features that they all share in common. And so we can infer that those features were also there in their last common ancestor. And we call this last common ancestor the Ur-Metazoan. But the same can be done for choanoflagellates. Choanoflagellates are actually very diverse, as you'll see in a moment. And if we compare amongst choanoflagellates, we can infer what was in place in their last common ancestor. And because choanoflagellates are the closest living relatives of animals, comparisons across these two lineages lead us to insights into their last common ancestor. Okay? So what is a choanoflagellate? How many of you have ever heard of one? Okay, good. How many of you have seen one? Yay! So I brought some cultures, and hopefully all of you will see them before the end of this course, but I just want to give you a little introduction here. So, the shape of the choanoflagellate is directly tied to how it functions in the environment. So this is the cell of a choanoflagellate. It's about 5 to 10 microns. It has an apical collar of actin-filled microvilli, and it has a long flagellum. And some people have described this as sperm with a skirt. Okay, so you can imagine the sperm cell, but it has this extra decoration. And the choano root is for collar. And so the way this works is the flagellum beats back and forth, and this creates water currents and pulls material out of the water column, primarily bacteria, but they eat viruses too. Okay, they eat anything they can get that's in the right size range. And so here's just an example in a scanning electron micrograph where you see the choanoflagellate, and just for scale, there's a bacterial prey that it's captured in its collar. So this interactions between choanoflagellates and and bacteria are very important. Now the shape of the choanoflagellate cell is very distinctive. In fact, it's not seen anywhere else in biodiversity except one place, and that's in animals. So only choanoflagellates in animals have cells that look like this. And this was first recognized by cell biologists in the mid to late 1800s. This is just one example of a comparison made by William Saville Kent. And you can see a bunch of, on this plate, a bunch of drawings of different choanoflagellates. They all have this structure of the collar and the flagellum. This we call the collar complex. And he noticed that the same structure also exists in sponges, where it's doing the same thing. The flagellum beats back and forth, and the sponge cells are also capturing material from the water column. And hopefully you can see this a little bit more clearly here if we compare the choanoflagellate cell to these sponge cells called choanocytes, now that have been imaged by scanning electron microscopy again, and you can see the cell body here, the collar of microvilli here, and then it's a little harder to see, but each of these cells has a long flagellum extending through the center of the collar. So choanoflagellates at a cellular level resemble cells from animals. And what I didn't emphasize is that they are actually as genetically diverse as animals. And so when I started studying choanoflagellates, it wasn't entirely clear which ones to study, because there was nobody actually really studying choanoflagellates at a mechanistic level, really focusing on particular species. And so this montage is just to give you some examples of all the different ways that they exist in the environment. So, you know, they almost all have a single-celled, important single-celled stage, and for some of them, that's all they do. But some of them can become multicellular. This is one example here. They form these spherical colonies. Some of them form chains. Some of them produce projections that allow them to either attach to each other or attach to algae or other things in the substrate. This is another example here. And then there's some really cool ones, which I hope we'll see, that secrete, they biomineralize silica and they make these baskets. You can see the baskets here, and then here's one where the basket has been stained in blue, and you can see the choanoflagellate cell in red. So there's a lot of diversity of choanoflagellates out there. And so it wasn't entirely clear which one to focus on, but I'm going to tell you about the one that and I'm happy to talk later about why we focused on this one. And so this is our model choanoflagellate, which we've named Salpingoica rosetta, or I'll just call it S. rosetta. And in the laboratory, it often is found like this, swimming around as single cells, each of those single cells living their happy life. But it was actually isolated from nature as a colony. And so this is an example of a culture in which most of the cells are existing in a colonial state. And you can see that these are roughly spherical, although they are definitely somewhat asymmetrical. But interestingly, all of the cells have their collars facing outwards, so there's some organization to these colonies. And these colonies develop, okay? They are not forming by aggregation. They're forming like animal embryos do, where there's a starting cell that divides over and over again. And so you can kind of see that here, the starting cell, it divides, and the polarity of the division is always the apical basal axis. So you divide into two cells with the same polarity, and then you keep getting these divisions, and they keep sort of curving around until at some point the circle is filled, and now the next cell division is going to be out of the plane. So that you get a three-dimensional structure. And so this movie starts with these two cells are going to become colonies. This one is already starting to become a colony. And so you'll get to see how this happens. I wish it were actually this fast. It takes about, each cell division takes about 6 hours. So it's a good 24 hours until you can see, bona fide colonies. But I think it's just a remarkable process. Okay, so just as if you're an animal developmental biologist, you want to understand how development is regulated. What controls the process by which you go from that starting cell to something multicellular? And we wanted to know the same thing. We wanted to know what what regulates whether this choanoflagellate is single-celled or whether it becomes multicellular. And I have to be honest, and I'm going to talk about this in more detail at the field station, but it was very hard to study this process. And the reason was that although the choanoflagellate was isolated from nature as a colony, in the laboratory it was almost entirely single-celled. And we stumbled on the answer, and the answer is partly why I was invited here. And the answer unexpectedly turned out to be that bacteria in the environment produce a signal that triggers the single-celled choanoflagellate to become multicellular. And it's not all bacteria, it's particular groups of bacteria, in this case from the genus Algoriphagus, which you can see here. It's a Bacteroidetes, and I'll tell you more about it later. But today I'm going to just boil down this finding and say that over a long period of time we were actually able to isolate the molecule from the bacterium that triggers this switch. And it turns out that it's a novel class of molecules called sulfonolipids. These are related to sphingolipids, which are important for signaling, and they're distinguished by this sulfonic acid head group. Okay, and this work was done in collaboration with John Clardy, and when I have a reference, I have it as a QR code here if anyone's interested. Okay, so we discovered that the choanoflagellate can alternate between being this single cell called a slow swimmer and this multicellular form that we called a rosette. But we've also discovered, in work that I'm not going to show you, that it can alternate to a different multicellular state called a chain colony. It can convert from being this big, happy, fat, slow swimmer to this small, streamlined, fast swimmer. This thing can land on the sediment, put out Philopodia, and convert into a different kind of cell called a thecate cell, which forms the stalk. And all of these can interconvert. So this is a very dynamic life history. This is an organism that can convert between many different cell types over its life history. And so that was fascinating to me, and I wanted to understand more about this. And one question we had, both for sort of purposes of discovery, but also for the purposes of trying to do genetics in choanoflagellates, was specifically, do choanoflagellates mate? And you'd think, I told you that choanoflagellates were first described in the 1800s. You would think that that would be known. But it wasn't, and in fact, it was thought that they were probably asexual because mating had never been observed. Now, why is this such a challenge? Why shouldn't it be obvious? I mean, I walked down the park, I could see dogs trying to mate, it's like you can tell what's going on. But the problem with the choanoflagellates is they don't make it obvious. So here they are, they're all swimming around. Every once in a while, you'll see a pair of cells, and you don't know, did they just divide? Are they encountering each other and fusing? And it's not really clear what to look for if you're going to look for mating. I'll speed forward and just tell you that a graduate student in the lab, Tara Levin, figured it out. And the problem is that choanoflagellates are quite shy. So she discovered that if she starves the choanoflagellates, they actually convert into the more heterogeneous cell morphology. So hopefully you can see that. But even when they're under this condition that we've discovered induces mating, the mating itself happens infrequently, and so it's hard to capture. So in this whole field of view, there are only two cells that are on the verge of mating, this little cell and this bigger cell. And because of the difference in size, we provisionally call this the male gamete and this one the female gamete. And so I'm going to zoom in on those two. There's the little one and the big one. And what you can see is that they sort of dance around to orient, and then they fuse. And under these conditions, you always see fusion between the little cell coming in at the base of the collar, okay? And so what this looks like is that under high nutrient, happy conditions, you're out at the coffee break level nutrients, they're all pretty much fat, happy cells, and they just divide, okay? But when they're exposed to decreasing nutrients, then what happens is that you start to get cell differentiation. When some of the cells become these little cells and they lose their collar, And we suspect that some of the big cells are actually not like the original mitotic cells, but that they're differentiating also. And so these cells encounter each other, and the little cell fuses to the big cell at the base of the collar, which I hope you can see here. These cells then fuse, and then that leads to the sexual cycle, okay? So when I'm representing low nutrients, it's gonna look like that sad, lonely pea. Okay, so another thing that happens when cells starve is that they actually swarm and they form these clumps. And this is going to be something I want you to keep in mind because I'm going to come back to it in a moment. Okay, so what has this done then? I told you that there was this wild and highly dynamic asexual part to their life history over here, and now we've discovered that they actually also mate. And so it increases the dynamics of this life history. Okay, so we were trying to understand in more detail how bacteria are controlling the life history of choanoflagellates. And we were focusing on the one I told you about, Algoraphagus. But we wanted a negative control, a bacterium that didn't induce multicellularity. And another graduate student in the lab, Ariel Woznica, was thinking about using Vibrio fischeri as a negative control because we knew that it didn't induce multicellularity. But when she added Vibrio fischeri, she did something that no one else had done before. Most people squirted it in, they came back 24 hours later and they said, are there any colonies? And there weren't. So we thought that Vibrio didn't do anything to choanoflagellates, but she actually looked right away. This is the, just because someone's done something doesn't mean that you shouldn't also give it a try. And so she looked right away and she saw something that no one else had seen. So this is the control on the left, control bacteria that don't, they're just prey, they're just being eaten. But when she added Vibrio fischeri in relatively short order, what she saw were these big clumps of cells, okay? Now at the time, we didn't realize that clumping was associated with mating, so we had no idea what was going on. We knew Vibrio was doing something, but we didn't know what it was. But then we realized that swarming is a common mating behavior in animals. Fish do it, bats do it, crabs do it, and these are these incredible lake flies in Africa that do it. And this is a very common feature across animal diversity, but also in the microbial world. And so we thought maybe choanoflagellates are mating too. And in fact, that's what's going on. So Vibrio fischeri, unexpectedly, it turns out, induces cell fusion and nuclear fusion in choanoflagellates. And so here now are two cells with their nuclei stained in magenta, attaching to each other. The membranes fuse, the nuclei now migrate, and they fuse, and you have essentially a zygote. Okay, and she did the experiments to show that in fact they were having genetic recombination and it was true mating. Okay, so when you get these really simple interactions between one bacterium, one eukaryote, that's a condition in which you can start to figure out what are the molecules that actually do the activity. And so I'm going to tell you this example of how we figured it out because it's so straightforward. So what Ariel did was to develop a bioassay for looking at clumping, okay, using clumping as a proxy for mating. So this is the control, and you can see that if she thresholds this image, she can see each cell, and she can measure the area. And if she does the same when she adds Vibrio fischeri, you get these clumps of cells, and she can measure the area of that and then plot it. Okay, so this is the area of the swarm for either control bacteria or Vibrio fischeri, and you can see that in the presence of Vibrio fischeri, the area is bigger. Simple, simple, simple. So then what she did was take Vibrio fischeri and fractionate it in different ways. And I'm going to just cut to the chase, and she found all of the activity in a protein, okay? And we've named this protein Eros, and you'll see why in a moment. But this protein, all by itself, we can add it, and now you get these big, big swarms, okay? So Eros, it turns out, is an enzyme. It's produced by Vibrio fischeri, and it's an enzyme that cleaves chondroitin sulfate. So animals produce a bunch of decorations on their cell membranes called glycosaminoglycans, and you've probably heard of a bunch of them: heparin sulfate, dermatan sulfate, hyaluronan, and chondroitin sulfate. And Vibrio fischeri produces an enzyme that they use to degrade chondroitin, partially as part of symbiosis, partially as part of essentially just eating, okay? And so we've named this molecule Eros after the Greek god of love, okay? So when we add Eros now, that is sufficient to induce swarming and mating in a way that's similar to what happens in starvation. There are important differences, but for the purposes of this talk, what I want to just say is that a signature of mating is this swarming and clumping behavior. Okay, one of the important differences is that the cells interact with each other differently, the two gametes, if they're induced by Eros, in which case it's the basal end to the basal end, whereas under starvation, you get these two different cell sizes, and they're interacting in a polarized manner, okay? So I have very quickly told you that parts of the choanoflagellate life history are regulated by extracellular cues. In the case of multicellularity, it's regulated by this lipid, and in the case of mating, it's either regulated by that enzyme Eros or by starvation. But what we haven't known is what's in the genetic makeup of the choanoflagella that allows it to perceive these cues. And so this next part of the talk I'm going to share with you is the work of a current postdoc, Alan Garcia de los Bayanos. And it's another example of he started out doing one thing and found himself studying something completely different, which was exciting. Okay, so he was interested in understanding what was the receptor for that lipid. And the best candidate for this is a group of proteins called G-protein coupled receptors, GPCRs. And these are really interesting, best characterized in animals. And one of the reasons why they're so interesting is that they can respond to a bunch of different extracellular cues. They can respond to peptides, they can respond to amino acids, they can respond to light, they can respond to lipids. And the GPCRs are the only known receptors for sphingolipids, which are related to that sulfonolipid that I told you about. And so he decided to try to study these in more detail. And he's done a lot of beautiful work on the evolution of this gene family, but what I want to do is zoom in on one particular GPCR that he's been studying. This is the GPCR, the spoiler alert, we've called it Cupidon. But other than that, there's nothing about it that would normally tell you this one should be particularly interesting. So it has a lot of diagnostic features of GPCRs. It has a 7-transmembrane domain. It has a site called a GPS, and this is an autocatalytic site that allows the extracellular portion to get cleaved from this portion. And then it has a bunch of relatively boring protein-protein interaction domains that are often involved in cell adhesion. And so he knocked this one out along with a bunch of other GPCRs. And in this case, he used recently developed CRISPR technology in choanoflagellates to cleave it either here or here. And every experiment we did, or he did, with these two different strains was the same. So I'm just going to focus on those with the shortest truncation. Okay, so the first thing is to show you, is it necessary for life in the choanoflagellate? And in fact, it's not. So the growth curve of the full-length protein versus the truncated protein is about the same. Here we're looking at time in hours, and here we're looking at cell concentration or density. And you can see that these two growth curves essentially overlap. So Cupidon is, they can divide perfectly fine with it or without it. Now, he was particularly trying to study it because he wanted to know whether it was the receptor for the multicellularity cue, which are called rosette-inducing factors, or RIFs, here. So These are cells that have not been treated with the inducer. These are cells that have been treated with the inducer, and they're either wild type or mutant. And what I hope you can see is that the wild type ones become rosettes in the presence of this factor, but the mutant ones do too. They look perfectly fine. And if we use that technique that I told you about where we measure the area, you can see that the full length, the cells with full length cupidon respond to rifts or the absence of rifts or the presence in the same way as the mutant cells. They're indistinguishable. So cupidon is not important for multicellularity. Sad. So why am I telling you about it? Well, because the knockout did have an interesting phenotype. And that phenotype is that when cupidon was knocked out, now suddenly all these cells start to aggregate. And so this was the first hint, and it led Alon to hypothesize that it might actually be important for regulating mating. And so what he did then was to measure cell fusion. And he did this by taking cells that were either expressing a green fluorescent protein or a magenta protein, mix them together, either in the wild type background or in the background of this cupidon truncation mutant, and then ask if they were incubated in high nutrient conditions, do you have cells with both fluorophores? And obviously you do, because we have a picture of it right here. But if you measure the rate of fusion, so here we're looking at the number of fused cells in the wild type versus the mutant, you see that in the wild type cells, under high nutrient conditions, you almost never get fusion. But in the mutant cells, now even in high nutrient conditions, when they shouldn't be mating, now you get about 0.5% of cells that are fusing. And this is just one snapshot. Over time, we think many, many more cells are fusing. Okay, so how then does this protein control mating? And I'll tell you that we don't really know the answer, but this is the first hint. He took that full-length protein and fused it to a little tag called an alpha tag. This can be detected by an antibody, and then expressed it in the coenoflagellates. And what I hope you can see, here's the flagellum and the collar. The protein is localizing at the base of the collar. And here is the collar stained and actin. And so we think that the localization of the GPCR at the place where the gametes fuse might be contributing to its regulation of the mating process. Okay, so just to sum up what I've told you then, we think that single-celled S. rosetta expresses this GPCR and localizes it to the bottom of the collar right here. Under high nutrient conditions, in evidence that I haven't shown you today, we know that this outer part of the protein actually gets cleaved off, and we think that that activates this protein and leads to a repression of mating. Okay, so because mating is repressed, these cells just divide and reproduce asexually. Now, in experiments that Alon did, if he keeps nutrients high, but now artificially gets rid of cupidon, it's no longer able to suppress mating. And so now these cells, it turns out, revert to a sexual life history. So it looks like you need to have active repression to turn off the sexual part of the life history, which I think is very interesting. And so what we still don't know, but I think will be very interesting to find in the future, is how does starvation impact on this process? Is it through, for instance, disrupting the cleavage process so that this protein is inactivated that way, or might there be new post-translational modifications that are getting rid of the ability of this protein to suppress the mating pipeline? But that's something that we're very interested in looking into in the future. Now, I want to summarize what I've told you, that, you know, I think one of the big take-homes that I want to give you is that this coenoflagella has a very dynamic life history that's regulated by the environment. And this is not true just for coenoflagella. This is probably true for all microbial eukaryotes in the ocean. And so, you know, we think of these organisms as simple, and I think bacteriologists have done a great job of really emphasizing the metabolic dynamics of bacteria. microbial eukaryotes are doing similarly diverse and dynamic behaviors, in some cases through differences in cell morphology and cell physiology. Okay, so that's one point I want to make. And the second is that this process of mating, which is really a process of cell differentiation, is regulated in choanoflagellates by a protein that's a homologue of proteins in animals. Okay, and through a coalescence of a lot of different lines of research, and I'll tell you more about that at Ecodim, we're now beginning to think of the transition to multicellularity as being sort of in the reverse order that we thought. So we thought that they had to become multicellular first and then have cell differentiation. But now what we're thinking is that cell differentiation is a fact of life for microbial eukaryotes. It happens all the time, it's integral to their life history, and the mechanisms for regulating cell differentiation were likely sort of co-opted into the multicellular developmental process. And so when we think about multicellularity, it's worth thinking about the dynamic life history of the organisms from which animals might have first evolved. Okay, and so I'm going to close by saying that we have a very friendly choanoflagellate community. We've grown. This was us in 2002. Here we are in 2019 in Barcelona. We had an online meeting in 2020 during the pandemic. You can't see everyone because I could only get one field of view. And we had another meeting in 2022, and we're having one this coming summer in Paris. So if any of you are interested in learning more about choanoflagellates, you can sign up for our Choano and Friends Slack workspace here. And we also have a website a spreadsheet that has contact information for all of the choanoflagellate labs and the labs of people who are studying similar research. And so we welcome anyone who's interested in learning more. And with that, I will take any questions. But from a student first, in any language of your choosing, as long as it can be translated into English.
Thank you very much, Nicole. Yeah, come on.
I teach undergrads. I can wait for a long time.
I'm sure you have questions.
Hi, Nicole. Thank you for your presentation. Very nice. I was curious about the Cecil one.
Yes.
Have you seen if they also form colonies or?
Yeah. So, okay. So this form, the thecate form. It's been harder to study. Okay. I think this is a really important point. We can study the transitions that we can regulate. So that sometimes we see cell types, we know that there must be a transition, but if we can't easily regulate that transition, it's harder to get mechanistic insight into. But in the case of this, we've started to notice that we most often see it after we induce mating. And so there's initially swarming, then we get cell differentiation, we get mating, and then suddenly we have all these thecate cells. And so what we think is that this might actually be the 2N stage of the life history, which would require us to redraw this. But it's been hard to really tease that apart. We have tried to add the multicellularity inducing factors to these thecate cells, and eventually they will become multicellular, but we think that the reason they're becoming multicellular is because they're releasing these slow swimmers first, and it's really the slow swimmers that are becoming multicellular, if that makes sense.
Yeah, thank you.
Thank you very much. More question?
Here's another student question. Oh.
Hello.
The real MC.
Yeah. I would like to know more about how you obtain and know that Eros was the principal structure that initiated all the process.
Yeah, okay, I'm going to talk about this in more detail at Queen A, but when you have a bacterium that you know has activity, you know it does something to your host of interest, but you don't know what the nature of the molecule is. You have some simple things you can do. So you grind them up. That's a good starting place. And you can then start treating what you've ground up in different ways. So you can boil it. That should destroy any protein activity. You can treat it with DNAs. That'll get rid of your DNA. You can treat it with RNAs. And you can also extract the lipids and try those. So if you do that with Vibrio, what you find is that boiling or treating with proteases gets rid of the activity. Okay, so that's the first hint. It's protein. Okay, so now how are you going to figure out which protein is it? Now, you have a few different ways you could do it. You could do a genetic screen and see if you could do a screen and find mutants that don't induce this, but that's not the most direct way to do it. We can talk about why that wouldn't be the ideal way. So what Ariel did then is she started putting the protein, so she extracted the proteins and she started separating them based on size. First big size chunks and then smaller ones, and she found that the activity was a particular size, okay? So she took that size range then and ran it out on a gel, and there were only a few proteins in there. Partly because I skipped over one part. You can look at the proteins that are still in the bacterium versus those that have been secreted. So it turned out that this one was secreted, and not a lot of proteins are secreted. So she took this size range of the secreted proteins, ran it out on a gel, and there were only like 4 or 5 different proteins there. So then she did two things. She took those, cut them out, and did mass spec, and she also cut them out and sort of soaked them out of the gel and tried that, and that actually worked for one of the proteins. And so that's how she figured it out. So it was one band, and then the question is that really it? So then she knocked it out in Vibrio, and in fact that got rid of the activity. And she did some really interesting tricks with editing the protein itself and getting rid of the catalytic amino acids, and that also got rid of the activity. So not only was it that protein, but it required the catalytic activity. So great question. Yeah.
Thank you.
And then I think there was another student question over here. Yeah. The realities of the hybrid presentation.
Thank you for your presentation. I want to know how long does the colony formations last, and what factors regulate it?
And say the second question again?
What factors regulate this?
If it
colony
reverts back to being single-celled, you mean?
Yeah.
Okay, so the first question is how long does it take to become a colony, I think, and that, so each cell division takes about 6 hours, and so if you just go 6 hours, you'll see cells that are next to each other, but you can't tell if they're a rosette or not. It takes about 24 hours to see a nice, good rosette. Now, if you keep the choanoflagellates in the rosette-inducing factor, they will just keep being rosettes. And you might think, well, does that mean they're just getting bigger and bigger and bigger? But they don't. They seem to have a size maximum. And then what happens is that the colonies either divide in half and split like that, or you have your colony, and what you'll see is individual cells kind of wriggling and fighting, and they like pull themselves out of the colony. And so as long as you keep them in that condition, they'll just keep being colonies. They don't stop. Okay, Now let's say you take your culture, you wash it, you get rid of the rosette-inducing factor. They will, as the cells divide, they will leave the colony. And so it takes some period of time for those colonies to stop, to essentially disintegrate or disaggregate. We don't know of a factor that we can add that will cause that. We just know that removing the inducing factor gets them to transition back to being single cell. Did that answer your question? Okay. Is there anyone who would prefer to ask their question in Spanish? I would love that. Maybe I'll even understand.
More question?
Thank you for your presentation.
Spanish, come on.
I
won't be able to.
I have a couple of questions. In the colony, I mean, are there colonies that are composed not by the division of the cells, but just aggregation of different cells?
Great question. So in this organism, no. If we block cell division, they will not become rosettes. Okay? But in another species that Josean studies, in fact, in a paper that's in review right now, they talk about the fact that the colonial form can form either through cell division or through aggregation. And this is a new kind of multicellular that hasn't been described before.
Right.
It has a lot of really interesting implications. So yes, there are examples where they form through aggregation.
Nice, thanks. The other question probably is a silly question, but these guys always have the flagella, or they can lose it, maybe during the division of the cell?
Yes. Oh, great question. Yeah, okay. So for those of you who don't know, it turns out that the structure that is required for spindle formation, pulling the DNA apart, is the same structure that is at the base of the flagellum. And so when this cell divides, it actually pulls back the flagellum, not completely, but partially. It then divides, and then the flagella regrow. Okay?
All right.
So that's during cell division. But there's another state that I haven't shown on here either that I need to add now, where it actually can convert from being a flagellated cell to an amoeboid cell, where it crawls around, and in that case also it retracts the flagellum, and then it grows back.
All right. Thank you.
You can also yell and I'll repeat your question if that's easier.
Vamos a practicar tu español.
Slower. How do I say slower?
Okay.
Vamos
a practicar, to practice tu español.
Yeah, I want to, but I guess not right now.
La mayoría de las células son N o 2N. 2N.
Right. These are 1N. Uno.
Una.
Uno N.
Y la meiosis, entonces, ¿cuándo ocurre? ¿Dónde ocurre?
When does it occur? Meiosis. Okay, good. So we have not seen it yet, but I mentioned that we think this might be the 2N cell. So we think this cell, someone has to help me redraw this, because we think this 2N cell actually goes here, and that meiosis might be happening here. So, and maybe someone will see it during this day. We have these cells, so you might actually be able to see it happening. Thank you. This is what I need.
Thank you for your presentation. It was amazing. I love that you have all this visual representation of what we normally see only in sequences.
Yeah,
like that. Do you want my question in Spanish?
If you say it slowly.
Es muy interesante lo que hace Vibrio. ¿Tiene algún beneficio para él cuando las células están en colonia?
What happens if Vibrio gets added to the colonies? No.
Almost.
There was a cuando in there, so how many? I don't know.
So if there, Vibrio induces the colony formation, right?
No.
Oh.
No. Okay. Okay, ask your question, then I'll, yeah.
Okay. So because of the induction of, we see that when you found the protein,
right.
I will, because maybe I not understand right.
Yes.
But I want to know if there was a benefit for Vibrio.
Okay, so let's, sorry, but I'll back up to that image. Okay. There it is. Okay, so Vibrio, I'm sorry I didn't clarify this. Vibrio induces swarming and clumping, so aggregation. So this relates to the question that Matthias asked earlier. These are not rosettes. This is a different kind of structure. In fact, it's not even a structure. You know, it's just a sort of loose association. Okay, so what is the benefit to Vibrio, and is there one? I don't think there's any benefit. So I like the notion of Estruzetta doing what's called hijacking or eavesdropping on these molecules that the bacteria are making anyway. Vibrio is making this protein because it degrades chondroitin sulfate, and that allows it to either invade, in fact, many bacteria make this enzyme, and they're all bacteria that are either pathogens or commensals with animals, because only animals and choanoflagellates make chondroitin sulfate, okay? So if a bacterium makes this enzyme, it is probably interacting directly with animals or choanoflagellates. Okay, so this enzyme is just being used to like shave off this layer on these cells, either to allow invasion or to eat, you know, to use it as a carbon source. That's all they care about. I think what the choanos are doing is they've learned that, they've evolved such that they are responsive to these molecules that are made by bacteria in particular environments. So the way I think about it is Vibrio thrives in an environment where it's adaptive for the choanoflagellate to mate. And the same with Algoraphagus, the one that induces multicellularity. I don't think the choano cares about the bacterium. I don't think the bacterium cares about the choanoflagellate. It's just that bacterium happens to be making this molecule, and that molecule is an indicator of a particular kind of environment. And that's something we can discuss, because I think there are these sort of passive cues. It's not that the choanos and bacteria actually even recognize that each other are there. It's just that there's this molecule floating around, and over time, the choanoflagellates that responded to that molecule in a certain way were more fit, and so they just keep responding to it.
So it is not necessarily causality, it is just co-occurrence of the
Yeah, I think so. Now, often I get asked, for instance, in the multicellular one, is that protecting the bacteria from the choanoflagellate? Does it mean that the choanoflagellate eats fewer of those bacteria? I don't think so. We don't see any difference in the prey capture rate. So we have no evidence that there's any selective benefit or adaptive benefit to the bacteria at all.
Thank you.
Sure. Audrey.
Yeah, so you mentioned that there is a huge diversity of kind of choanoflagellates. So does the same type of behavior and cycle been observed in other choanoflagellate species or branch, or is there any diversity of behavior that has been observed?
Incredibly diverse. In fact, with any given species, the closer we look at it, the more dynamic we appreciate its life history is. And so far we have, for instance, when we add RIF one to other species that we know can become multicellular, they don't, which says that they're responding to different cues. And the same, if I'm correct, and I'm going to look to Hosean for, I think it's true. When we've added arrows to other koanos, we don't see mating, even though we know that they mate. And the species that Hosean studies, the C. flexa, has a very wild life history, but it's completely different from this one. So I think that this is a case where we're just scratching the surface. We only know about a few choanoflagellates in detail, and the more species we study in detail, the more we're going to see these dynamic life histories. And I should say, one thing that's been interesting is that if you look into the literature, there are often, pictures or sketches of colonies, they get one name, and pictures and sketches of other single cells, and they get a different name. And now we've discovered that, in fact, they're the same species, just different life history stages. And so we might have to completely reorganize the way that the naming structure of choanoflagellate diversity.
Thanks, Nicole, for the really interesting presentation.
Thank you.
Now my question is, if I understood correct, when you add arrows and when you have starvation,
yes,
you have mating.
Yes.
But the starting point of the mating, the cells have completely different morphologies and the way they interact as well. Right?
Exactly.
So my question is, the offspring has any sign that has occurred? Like they have any differentiation, or they have a different behavior, or the genomes, I don't know, get somehow messed up, I don't know.
Right.
Is there any difference or no?
At the level of the genome, we don't see any difference. So they just recombine. And we have strains that have different genetic markers, and we can induce mating, and we just see random assortment of alleles in the progeny. Whether there's some difference in the cell state of the offspring, we don't really know yet. And partly that's because we don't have, if you do a yeast tetrad analysis, you get mating, you get meiosis, you have the 4 offspring, and you can study them because you have them on solid media. But with the koanos, they swim around all the time, and it's hard to capture individual cells. And so we're still trying to figure out how to do that. It's a great question, but we really have no idea.
Okay, thank you.
Thank you very much. I don't know if there is another question. I want to ask you something, because at the end of your presentation, you mentioned that the differentiation and the multicellularity perhaps are the opposite, that is like the most logical. But then I remember Caulobacter. And this kind
You remember what?
Caulobacter is a kind of bacteria.
Caulobacter,
yes,
yeah.
That's so
exactly.
And do you know if there are some molecular evidence of that can be similar molecules interacting on those processes?
They're completely different. Caulobacter, the genetic underpinning is completely different, because they're using like histidine kinases to regulate that.
Okay.
And it's a completely different genetic pathway, but I think it's I think bacterial development and cell differentiation is a good way to think about sort of the fundamental mechanisms. Exactly. It's just that the specific genes that are involved are just completely different. Yeah.
Okay. Thank you very much.
Yeah.
Do microbial eukaryotes have polyploidy? Are they, or?
Yes, some of them do. So, like, diatoms are just awful. They just have the worst. In fact, it took a really long time to get diatom genomes sequenced, because the first species they went after were horrendously polyploid. In terms of koanos, so far they've all had really boring, and pretty much 1N genomes when we grow them. It's hard. Those 2N states are transient, So they're pretty much just one in. They look like animal genomes. They're just, you know, they have coding sequences. They do have introns and exons, but nothing particularly interesting in terms of their genome organization.
And do they do like crossing over?
Yep. Yeah, they do crossing over. We see that as the you know, recombination and the offspring of mating.
Cool.
Yeah.
Thank you very much. Okay, you made it.
Yay!
Full of questions. Thank you very much. I think we are ready for now. We invite you to the lunch time now at top of the hill. We will walk together, and then we come back at, I don't remember, 2:13. 2:13. Lunch? Okay. And then come back at 2:30. Yeah, exactly. So thank you very much. Perfect. Good afternoon. We are coming back to our last part of this wonderful, big, huge symposium. Our next speaker will be presented by Mariana Rodriguez from Universidad de Sao Paulo, I think, yes? Following this tradition that we will implement now, that is a national introduction.
Good afternoon, everyone, and thank you for joining us for the 12th edition of Ecology and Diversity of Marine Microorganisms course, Ecodim. My name is Mariana Santos. I'm a PhD student at the Oceanographic Institute of the University of São Paulo in Brazil, and it's my great pleasure to introduce our next speaker, Dr. Adriana Lopes dos Santos. As a Brazilian people say, carioca da gema, she is an incredible marine microbial ecologist interested in understand the structure of marine eukaryotic phytoplankton communities, and just like me, how the environmental factors can shape their diversity, combined traditional culture isolation and laboratory studies, along with cutting-edge DNA sequencing approaches to elucidate the patterns of marine eukaryotic phytoplankton across a wide range of environments. She had a bachelor degree and master's in genetic and molecular biology and a PhD in environmental biology from the Federal University of Rio de Janeiro. During her PhD, she was awarded with a government scholarship that allowed her to spend a year at the University of Michigan in Dr. James Tidal Laboratory. She was fascinated by the marine microbial life and started this academic way as a junior postdoctoral research fellow at our academic home, Oceanographic Institute of the University of São Paulo, when she attended as a student at Ecodim and Seymour in Hawaii. By the 2012, she moved to France, where she worked for 5 years at the Station Biologique in Roscoff, focusing on marine microalgae connecting large-scale molecular biology datasets with classical methods. Between 1218 and 1223, she was a professor at Nanyang Technological University in Singapore. In 2023, he moved to the University of Oslo in Norway, where she is currently a professor at Geek Lab. And today, she's going to present a talk, Diversity of Microbial Plankton in the Southwest Pacific Ocean, a synopsis from carbon production to export. And as a good Brazilian way, vai que é sua, Adriana.
Thanks, Mari. Okay, so good afternoon, everyone. So as Mari said, I'm Adriana, and I'm the lead PI of the Genomics and Ecology of Eukaryotes Lab, so the Geek Lab, which has moved now to Norway. But I'm also an Ecodim alumni, so I was here in 2012 as a student. I'm not proud of my post, but we are still friends here, so this didn't affect our friendship during Ecodim. And I'm here back in Ecodim in 2014. So be here has a very special place for me, not only because I was part of this course, but also because the idea of what I'm going to present today started actually here. So when I came in 2014, and as a TA, I had to give a class. And during my class, I was showing the distribution of different phytoplankton groups in the global ocean. And I was showing that some groups was highly abundant. And I was very proud of the result. It was fresh out my computer, and I was giving my class. And then at the end of the class, Osvaldo raised his hand, looked at me and said, well, that's all interesting, but what's the participation of these groups in the carbon cycle? And that stuck to my mind. I didn't know the answer. So now I know a bit more about the answer, and that's what I'm going to present here today. So it's a project that actually started the idea there. I applied in Chile for that project. The first experiments we did, we failed miserably, and then we tried it again, and then is what I'm going to show you today. So that's why I choose this topic, and as I said, for me, it's very special to be here. So please interrupt me during the talk if there is anything you don't understand, or if you want to ask questions in Spanish, is fine. I'm going to answer in Portuguese. In Portuguese is also fine. So. Okay, so I work with marine plankton. Here in this video you can see that there are different types of cells in the marine plankton. Plankton comprises all the organisms that are drifting in the water column, and they cannot swim against the current. That's the basic definition. But in the plankton realm we can find different cells with different lifestyles and also different groups. So plankton communities are taxonomically diverse. They have been historically divided based on the size, and this leads to a very broad classification of this community into what we call picoplankton, nanoplankton, micro, all the way to megaplankton. So just to remind you, if you're not familiar with the plankton, so for example, the difference in size between a picoplankton cell and an organism in the mesoplankton can be similar to an insect, to an ant, to a whale. That's the range of size spectrum that we are talking here. Also, the plankton communities are taxonomic diverse. What I mean by that is that you can find all the 3 domains of life. So you can find viruses like Matthias showed this morning, and we're going to come back to that, bacteria and archaea, and eukaryotes. So within eukaryotes, we have the multicellular eukaryotes, so that's like jellyfishes and other organisms as well. But also we have unicellular eukaryotes, so called microbial eukaryotes, like choanoflagellates that Nicole showed us just before lunch, that are mostly present in this size fraction, nano, micro, and picoplankton as well. So you have all the 3 domains of life, all these different size ranges as well. Besides these, plankton communities are also functionally diverse. And you can classify these organisms based on the way they acquire resources. So one that we are more familiar with, the 2 that we are more familiar with, are the phytoplankton. So this comprises the cells that are photosynthetic, so they can do photosynthesis. They are constitutively photosynthetic, like the plants in the land. And then within the phytoplankton, you're going to find bacteria, so the prokaryotes, the cyanobacteria, and microbial karyotes. So, and within microbial karyotes, photosynthetic microbial karyotes, you're going to have different lineages as well. And the zooplankton. So the zooplankton are the heterotrophic organisms, so they will acquire the nutrition or the resources by prey, actively prey, by mostly heterotrophy, so phagotrophy. And you can find within the zooplankton both uni and multicellular eukaryotes. So one thing I forgot to say, sorry, in this diagram we have all the different functional types. So for phytoplankton, coming back, we have diatoms here, for example, Chetocerus, which is a microbial karyote, and then you have Synechococcus here, which is a prokaryote, a cyanobacteria. On the zooplankton, if we look unicellular zooplankton, we have, for example, ciliates here, while for metazooplankton, so multicellular, you can find copepods, you can find also larvae of barnacles, jellyfish. So you have all these different types of organisms within the same functional group. But one thing that's special about marine plankton is that there is another form that's becoming more and more known now, which is the mixotroph or the mixoplankton. So this is organisms that can
combine the two different types of resource acquisition. So they can be autotroph, and they can be permanently autotroph, or they can, like for example, carnivorous plant on the land, they are phototrophs, they are plants, but they also can acquire nutrients by prey, by eating insects. The same occur in on the plankton realm, so you can have organisms that are constitutively autotrophs and then can perform phagoheterotrophy when the conditions are changing. So in this diagram, for example, we have a group here, the haptophytes, we have some ciliates, and we have dinoflagellates. And we will see this through the course in more detail of this group. So this is the mixoplankton. And now we know that they are actually more abundant than we thought before, and their role in the marine ecosystem is even more important than initially thought. So what's one of the major role of microbial plankton? So you saw a similar diagram. This is a food web diagram, but also a representation of one of the most complex food web we have in our planet, which is the marine biological carbon pump. You saw this in the presentation of Matthias, and he got your attention of how carbon can be transported or generated by the activities of the viruses. So the marine biological carbon pump is a very complex process, a very complex food web that guarantees that the carbon that is fixed through photosynthesis here on the upper layer by phytoplankton and mixoplankton is transported all the way down to the ocean. And this is actually done by different organisms, and we will see in more details. So what's the impact of the biological carbon pump? One is the long-term carbon sequestration. So in any conditions, the CO2 that's in our atmosphere will dissolve naturally in the water, right? But if the concentrations on the surface is high of CO2, if that CO2 is not transformed into organic carbon, so the concentration is reduced, or transported down, this dissolution will not happen anymore. So by actively doing photosynthesis in the upper layer, both phytoplankton and mixoplankton, and by the transportation by the other organism, the biological carbon pump plays an important role in the long-term storage of carbon or sequestration in the deep waters. It's also the source of inorganic and organic nutrients, and this happens in the more batch pelagic layers through the transformation and remineralization of this material by bacteria. And finally, the biological carbon pump is also fundamental for the functioning of the marine ecosystems. Both batch pelagic and mesopelagic and benthic ecosystem rely heavily on the sinking of the organic carbon that's produced in the initial surface layer. So it's a very, very important process that guarantees the function of our ocean, but also that controls how much carbon is getting taken from the atmosphere. But despite of the importance of the biological carbon pump in our planet, there is our understanding of the efficiency of this process across different oceans remain very limited. So in this graphic here, we have 4 different Earth system models, which is heavily used to assess how much carbon is being taken by the ocean. And one way the efficiency of the biological carbon pump is measured is by how much carbon has been transferred from the surface layer to the ocean, okay? Especially at 1,000, 2,000 meters. So as you can see, this is a models that estimate the efficiency of the biological carbon pump. And they are completely different. They give completely different results. And the input information here is the same. And one reason for that is that we don't know how the different groups in the surface layer are producing, what's the rate of they are producing organic carbon, that they are fixing carbon. And also we know much little about when carbon is transported down in the water column, what's happened, what kind of transformation, what kind of losses is happening, and how the community in the surface layer influence that at the depths. So those are holes that we have on understanding the biological carbon pump, and that's where we're going to try to get more. So coming back to our drawing of the biological carbon pump here. So in this talk, in the first part, we will look how carbon fixation rates by small phytoplankton groups change in different oceanic conditions. So we're going to look primarily on primary productivity here. And in the second part, how changes in the particle-associated microbial eukaryotic community, so the community that's associated to the particles that are sinking, are transformed and changed during the export, and how this affected what actually ends up in the sediments and in the bottom layer of the ocean. So to answer, to tackle these questions, we collaborate with the National Institute of Water and Atmosphere in New Zealand, here. And we went to this region, and the Chatham Rise region. So here in this graphic, what we have, the color represents the temperature of the surface of water. This is an average of a month. And the lines is the bathymetry, so the depths. So, and what you can see in this region is that in the Chatham Rise, which is a shallow region on the Southwest Pacific Ocean, there is a mix between these water masses that are more, that are warmer and also nitrate limit. So there is a limitation in the growth of phytoplankton by the low concentrations of nitrate, with waters that are coming from the subantarctic layer, which these waters are rich in nitrate but have low concentration of iron. Iron is a micronutrient that's very important also for production, for fixation of carbon. So because this region is shallow, there is a mix between these water masses that promotes production by small phytoplankton. So that's where we went, and why it's interesting, because we could sample this water mass, perform experiments in this water mass in the same voyage. So you don't have to go so far to find very contrasting oceanic conditions. We want to test how the populations in these water masses and also in the region where they mix were fixing carbon, and how this was influencing also the transport of carbon. So during this voyage, we sample in a different style. It's called the semi-Lagrangian style. So you are following the same water mass, or you try to follow the same water mass, the same population through days, okay? And in each one of these sampling cycles, so this is a 3 or 5 days that you follow the water mass, that you drift, we perform the experiments and we collect the sample, which is here represented by these points. So, coming back to our first question, so how does productivity and taxonomic diversity of small phytoplankton population have changed across these nutrient limit conditions, okay? One rich in nitrate but limited in iron, and the other one deficiency in nitrate. So what do I call small phytoplankton group here? So in this case, we are looking into both eukaryotes and cyanobacteria. Cyanobacteria, we're going to be looking to one genus, and in this case, Synechococcus, which is the most prevalent in these waters. But when we look into the other size fractions, for picoeuc and nanoeuc, and here the size, we have different lineages of eukaryotes, of microbial eukaryotes, that are slightly larger. And here are they. So the most significant groups that we found, that we find in these of phytoplankton, so photosynthetic microeukaryotes, in these size fractions are the diatoms that you are very probably well known, green algae, within prasinophytes, dinoflagellates, and haptophytes. And within each one of these groups, there are several lineages that are genetically distant between them. So why we are looking to small phytoplankton? In this region, not only in this region, but small phytoplankton dominates oceanic primary productivity. And that was where the question of Osvaldo was coming when I was a student. On the top, we have chlorophyll. We have a model of global chlorophyll concentration through a year. So as the color increases to more green, you have more chlorophyll, means more biomass, means more phytoplankton. And on the bottom, the colors represent the different small phytoplankton groups. So more red, bigger they are. More green, smaller they are. They are within the pico and nano size fraction. And as you can see, there are regions of the ocean that's completely dominated by these small cells, where productivity is dominated by these small cells. And that's where we are looking for, exactly here. So what do we did in this first part to address the question how production changes across the different groups? So we collected the water in different depths. We brought back to the boat, and we spiked with bicarbonate. And bicarbonate that was labeled, radio labeled, so the carbon is a radio label. So we can trace what's happened to the carbon as the groups are fixing. And then we put it back at sea at the same depth that we collected the water. And then we pray. You wait 24 hours for your experiments that are drifting, and with the hope that you're going to recover all your bottles and be able to do your calculations. So back on the ship, back in the lab, we use an equipment, a flow cytometry, to separate the cells in different groups. So the cyanobacteria, the Synechococcus, the picoilk, and the nanoilk. And this is based on the size, but also the type of pigments these cells have. And on the same sample, and here is where we innovate, in the same exactly sample, we measure how much carbon was fixed, and we look who was fixing, which community was present in our incubations, by using amplicon sequencing techniques. So you amplify a small gene marker to try to look who is there in your community. And we use different markers for both the Synechococcus and the pico and nanoeukaryotes. So, okay. So let's take a look into how the rates per cell are changing between the different water masses. So in this graphic, we have on this axis the different populations, Synechococcus, picoeukaryotes, and nanoeukaryotes, and here carbon fixation rate per cell. And the color is the different water masses. The violin plot shows the different measurements that we have. We had for each experiment, triplicate, several experiments per cycle, etc., etc. And the point here is where I want you to look, is the least square mean at 95% of confidence, meaning that's where we are looking in terms of global production for each one of these groups. So the first thing you can see is that Synechococcus in general have a lower rates compared to picoeukaryotes and to non-eukaryotes. This is well known. This is like when you look your experiment and you see a results that people have done before and say, whew, I did good. It's okay. I'm in the good track. So, but when we look across the different water masses, we see that Synechococcus have a lower rate in subantarctic water compared to subtropical waters, although the community that is there seems to be adapted to their conditions, and I'll go back to that. But what got our attention is that both non-eukaryotes and picoeukaryotes, there is no statistical difference between these rates in the different water masses. So despite the different nutrient conditions, the community within these two groups are performing similar rates of carbon fixation. So small eukaryotes here, both pico and nano, have similar productivity in both subtropical and subantarctic waters. So we went to see how this is compared to other studies. There are very few studies that did this kind of measurement, and that's because of the difficulty of this kind of experiments, and most of the experiments have been done in tropical waters or subtropical waters. So here we have the different studies starting from 1994 to our study. the different groups, Necococcus, Picocaryotes, and Nanoeukaryotes, and the colors represent the different water masses. As you can see, most of the previous results are in subtropical areas. So for Synechococcus, this is our first recording, the lower rates of production for Synechococcus in cold waters. But what's important here is that our rates, for example, for nano, were not different of the only study that also looked into nano eukaryotes production rates. It was actually very similar. And for pico, we are also at lower rates compared to some of other studies. So one clear trend here is that bigger you are as a cell, higher is your rate of carbon fixation. But that can be because the rate is scaled to the volume of the cell. It doesn't mean that the cell is dividing faster, it's being more efficient in capturing that carbon. Okay, so that was one question that we had. So we use the data from the carbon fixation and the biovolume. Yeah.
So I don't want to interrupt you. You want to finish your thought?
Yeah. Okay. So we use the rates that we had and the biovolume we acquired from the flow cytometry to see the growth rates of the cells. So how much the cells are dividing in the environment.
So when you're measuring carbon fixation per cell,
yeah,
do you have a way to make sure you're only measuring it in the phytoplankton?
Yeah.
Or
Yeah, because they were sorted.
sorry.
Because the cells are sorted, so I'm assume that the majority of them has chlorophyll and a phytoplankton.
And is there any chance you're measuring small phytoplankton that emit?
Yes, there is a chance. The thing is that we also, in our experiments, there is a control. And I would expect it that if that is happening at the higher rates of consumption in the 24 hours of experiment, so the control is that you perform at 12, you perform at 24. If there is a change in the 24, you have a higher graze happening there. So also the cells, once it's grazed, I would expect, since this is per cell, that the signal is lower than when I'm sorting the whole population as a, yeah. I don't know if it makes sense, yeah. Okay, so coming back here, so this is the growth rate, the estimated growth rate from the carbon fixation. And what we see here is that there is, although pico and nano are fixing at the same rate, in subantarctic waters, nanoeukaryotes have a high growth rate, so they are more efficient in capturing carbon in these waters than compared to pico-eukaryotes. Okay? So nanoeukaryotes are more efficient in carbon uptake compared to phytoplankton in subantarctic waters. In subtropical, actually pico-eukaryotes seems to be more efficient than nano. Okay, so there are differences in the way these groups are behaving in the environment related to primary production. So, but who is there? Because we sequence who is performing those rates. So who is there? So here we have heat maps. The lines are the different taxonomic groups, and the columns the different samples. So as I said, for Synechococcus, there is an increase in subantarctic waters of this clade, which is known to be adapted to low iron concentrations. So although they are adapted to grow in their conditions, the performance in carbon fixation is not as good as pico and nanoeukaryotes. When we look at pico-eukaryotes, there is an increase in subantarctic waters, in iron limitation waters, by some primizophytes, pelagophytes and chrysophytes we're going to see, but there is a differences. In subtropical, you have more green algae, while here you're going to have other groups in subantarctic water dominating. When we look in nanoeukaryotes, there is no clear pattern. There is a dominance of dinoflagellates across the different water masses, and also the presence of some of these haptophytes. So we were like, okay, what they have in common, this group. So why these pico and non-eukaryotes didn't show difference in the rates in the different watermelon? What could explain the adaptation to different nutrient conditions? So we look for traits that could explain that. So we took the sequence, we took the taxonomic annotation, and we tried to infer traits to that annotation. And what we found by looking database and also looking through the literature is that there is an increase of groups that are performing phagomixotrophy. So as we go to subantarctic waters, both in the pico and in the nano is from subtropical to subantarctic waters, we have a dominance of phagomixotrophy in the population that we are looking. Here, I'm not saying that we saw this group doing phagomixotrophy. We could not see that, but that's what's indicated, and then opens the door for testing how much that's happening in the field. So here we suggest that this resource acquisition, this functional group, is allowing this pico and nanoeuc to adapt to both iron and nitrate limit conditions. The same has been shown for phosphorus as well, for limitation in phosphorus. So to go back to our major questions, so we look how it's produced. Now we're going to look how the community affect the transport of this carbon to the deep layers of the ocean. So here we are back to the carbon, biological carbon pump. The way carbon is transported from the up layer, as I said, to the bottom, it can be done by three different ways. So you can have the physical mixing. So as the concentration decreases at the surface, by mixing of the water column, dissolve inorganic carbon that's in the bottom layers goes back. You can have also vertical migration of the organism, so you can have zooplankton that during the night go up, feed on the phytoplankton that has been growing, and then go down, bringing the carbon down. And you can have the gravitational sinking of particles. So this is the formation of particles of dead cell, fecal pellets, etc., that is sinking from the surface to the bottom. And that's considered one of the main processes contributing to how carbon is channeled down to the deep layers of the ocean. So, but when we talk about gravitational sinking particles, like Matthias presenting his talk, we need to be aware that there is also a diversity of these particles. And that means a diversity of the community that's generating and that's residing in these particles as well. So you can have aggregates, for example. You can have fecal pellets that's produced by zooplankton, by fishes as well. You can have also cells, phytoplankton cells, and even some mixotrophic cells that are large enough to sink. Okay? So you have all these different types of sinking particles. And the community that's in the surface will determine or will affect highly how these particles are generated. So for example, if you have a community that's highly dominated in zooplankton and copepods, you're going to have large aggregates that are more dense and sink fast. Sinking fast means you are less prone to change as you are going down, because you are going fast. If you are more porous, for example, communities that are highly dominated by small cells, you are more porous, you are small, you sink less, the speed that you sink is lower, so you are more prone to the bacteria and the community at bathypelagic layers to work on the material. So therefore, the community at the surface will affect the type of particle generated and also the fate of this particle. So going back to where we are, in the Chatham Rise, we have again the different types of water masses, and we deploy different sampling strategy to try to look how the particle, the community associated to the particle is changing, how this affected the fate of the cells down to the deep layers. So we have traditional kind of sampling of the upper layer. We deploy large water pumps to the deep layer to look into the reside community of both pelagic and mesopelagic, and we deploy what's called the sediment traps. So this is a traps that you put, this is a cylinders that is filled of formalin that you put just where you want to capture these particles that are going down, that are sinking. So you leave there for a couple of days, and then you will cover your material, hoping that the particles that are sinking will get trapped by these cylinders. Once the particle gets inside of the cylinder, since there is formalin, the community is killed. So any transformation of the particle will stop. So this is called the particle interceptor traps or sediment traps. And of course, we sample also the sediments. So our objective here, considering that there are differences in time that this process will happen, is to look how the cells that were here will end up here, basically. So that's our question, okay? So we use different methods to analysis. So we use indicator species to say this is a species that reside in here. This is an indicator species of the surface community, and then we look at how the abundance of the given species or the sequence that show the presence of the given species will change through the water column. What do I mean by that? So how we evaluate the potential of transfer of the microbial community from the surface. So here I have my traps below the surface water column, so below the photic zone, and I have lower traps in the mesopelagic, and I will have my collection of sediments. So if a given taxa, for example, increase from the surface to just below the mixed layer here, I say they have a high export rate, or that's how I look export. If a given group is transferred from the below the mixed layer down to the sediment, I'm looking to that transfer, okay? So let's see some examples with the cells. So let's say I have two species. I have species A and B, and they have similar abundance in the surface layer. So here I have a green algae and a ciliate, for example, a microcaryote, okay? So they have similar concentrations at the surface. So just below the mixed layers, just below the photic zone, I see that species B abundance have been reduced, while species A remain the same. So I say that this species here has a high export, and this one has a lower export. So it's being less moving down in the layers of the ocean. But then when I look at the sediment, I see that the abundance of this species remain the same, while this one has reduced. So I say that there is a lower transfer to the sediment, and here there is a higher transfer. So what that means? It means that this guy here, for example, was probably consumed all the way here in the batch pelagic, and very few cells have reached the sediment. That's one hypothesis, okay? While this one, which have a low export, they will probably consume here in the upper layer, for example, by different type of grazing, and have been channeled out all the way, for example, in fecal pellets, okay? That's one way of one hypothesis. And then you can have, of course, species that have low export, lower transfer, and high export and higher transfer. So the concentration remain high through all the different layers. So that's how the data look like. And after lunch, and after listening to me for almost, I don't know, half an hour, 40 minutes, I'm not gonna stay on this graphic for long, but that's an example of subantarctic and how we look the different abundance of the different groups of microbial caryotes, and how we determine if they had low export or higher transfer, and high export and low transfer, and etc. Okay? So to summarize, what we found was this. So for some groups, for example, for radiolarians, in subtropical waters, what we had is that this group had a high export and higher transfer. So we could find the highly abundant all the way from the surface to the sediment. And this is exactly the same sequence that we are tracking. It's not just the taxonomic group, okay? While in subantarctic waters, radiolarian had a high export, so in the mesopelagic layer, they were high, still high concentrated or high abundant, but in the sediments, they were nearly absent. So for this particularly group, depending on the water mass, you're gonna find different behaviors or different contributions to the export of carbon. Ciliates and all the other groups, independent of the water mass, they have the similar behavior. So ciliates, both in subantarctic and subtropical, had high export and lower transfer. So they were not found on the sediment, although they were highly abundant in the mesopelagic layer. When we look low export and high transfer, we have the diatoms, the traditional groups that have high export that we know. The high transfer, sorry, that we know, but we were surprised to see some of the green algae like Chloropicophysia and Prasinodermophyta that tend to disappear on the bathypelagic and mesopelagic, but has a high accumulation in the sediments. And when we look into traditional groups that have been reported in subantarctic waters like Phyllocystallis, what we found is that they have low export, but have higher transfer as well. So they are accumulating in the sediments. So this is just an example how these different groups have completely different behaviors when it comes to export. Do we know why? No, we don't know. We can make hypotheses linked to the cell morphology, the type of cell cycle that they have, and also the interactions that they have with other organisms. So like what kind of grazes they have and how they can end up being consumed in the bathypelagic or in the sediments. But besides that, we don't know. But now we can look into those questions. We can look at how these different groups behave concerned to carbon export. So, message from here, export patterns for taxon remain the same across different water mass except for radiolarian. And what we propose is that we need to consider some traits, specific cell traits, when you are looking for the contribution of certain groups when it comes to export. So as take-home message, first, from the first part, small eukaryotes had similar productivity in both subtropical and subantarctic waters. Phagomixotrophic of small eukaryotes might be the strategy to overcome nutrient limitation in both nitrate limitation and iron limitation conditions, limit conditions. Most of the surface diversity was lost during export. I didn't show that, but most of the diversity was highly consumed at the upper layer. Finally, a higher proportion of green algae belongs to these groups were exported to the sediments, which has now been reported before. And what was the most new or that caught us by surprise is that the exported pattern differed between taxa but not between water masses, which indicate that the cell traits here plays an important role in the function of these groups. So this was done in collaboration with a lot of people, but I want to call your attention to Denise, which was a former PhD student in the group, which actually is the one behind everything I showed you here, except some of the experiments that we did. Also now the Geek Lab that has moved to Norway is freezing up now in Norwegian waters. The great help we had from NIVA to perform these experiments, and also the support from Roscoff Culture Collection, Dominique Marie, which some of you know here, which actually help us with all the sorting and all the processing, especially of C14 samples in a flow cytometry. Not everybody wants to do. So thanks to them, I'm able to present this work today. Questions?
wonderful talk from Dr. Lopez dos Santos. Do you have questions?
Ooh la la, here comes my second project. You?
Ah, vamos.
Pode fazer em português, pode. She will do in Portuguese and then we translate.
First of all, I'd like to thank you for your talk. E a minha dúvida é se você acha que, por conta dessas mudanças climáticas, o que você espera alcançar e quais foram as dificuldades que você encontrou ao longo do processo com essa abordagem que você fez no trabalho.
Speak here. Yeah. So the question is with the current climate change, what I expect from what we saw, and also the challenge of doing this kind of work. So I'll start by the challenge, that's the easy one. So the challenge is to perform this kind of incubation in situ. We had, you lose samples also to work with carbon-14. It's not really well received in many places. But also if you look the kind of studies that have been performed before, I'll go back here. There is nothing, if you go up, for example, to subarctic and Arctic waters, or even Antarctic waters, and the reason is the number of cells. So when you are sorting to do cell-specific rates, you need to have a number enough that you are able to quantify, and that's a major challenge as you move to, if you are working with eukaryotes. So with bacteria is different, but if you are working with eukaryotes, so that's some of the challenges. That explains why there is so few similar work. And also for the traps is to have that kind of sampling strategy is not easy as well, so you have to have a great support. For the climate change, I'm not a modeler, but so one thing that we want to do now is to apply the rates that we found in the current models and see, especially with the expectation that the population, the phytoplankton population will become more into the piconano in regions that are not dominated by piconano, and see what's the result. So that's one thing that we are planning to do now. But with colleagues that are Earth system modelers, so they can apply the rates that we found. For the traps, it becomes more complicated because trade-based ecology is something that's not really solid, I would say, in the, we are moving, but so if a modeler asking me what's the trade do you think is the most important, it's hard to answer that question right now. So, but we know that there are differences, so that's already one step, I think. Yeah.
Thank you. More question?
Nicole.
Go.
The next student who asks a question, I'm taking you out for a drink. So. Okay, so I have two questions. One is, how do phototrophs do heterotrophy? Are they doing phagocytosis?
Yeah, in this case, we are using the definition by phagotrophy, because right now we consider that by osmotrophy, most of the phytoplankton are osmotrophs. So that's not the kind of phagomixotrophy we are looking here. So when we look at the literature, we look either for evidences of species that have been doing phagotrophy in the lab, also genes related to phagotrophy for some of the haptophytes, for example. So that's how we look and try to focus only in phagotrophy, phagoheterotrophy. Yeah.
So I'm just having trouble picturing. I mean, I don't know how it works, but are they breaking down their cell wall?
Not all of them have cell walls, but the mechanism is not known for many of them. Like for dinoflagellates that have a theca that cover in a hard shell of cellulose, I don't think we know how they do phagotrophy. Yeah. So for many of them, we don't know the mechanism.
Maybe we could look.
Mabel?
We could look during this.
Absolutely,
wonder
I would
if
love
that would
it.
be fun. And then the other question I have, because I don't know this technique very well, so you're sorting,
Yeah.
and then are you doing the carbon measurements on a cell by cell basis, or is it on a bulk?
In a bulk.
I
bulk
see.
of population. So cell by cell will have to be like nanosim. But then you even there you average. So here you sorted a group. You sort a number of cells, and then you measure in that how much carbon has been accumulated in there. And since you have the number of cells, you kind of estimate per cell. You have the size, volume, and you kind of estimate per cell.
I have two questions. Thank you for your presentation. And the first question is, why you don't include the microphytoplankton size in your research? The microphytoplankton size.
What I have done with the bigger cells, the bigger
Exactly.
cells. Okay.
And how you can include the abiotic factors in the export of carbon, for example, in different depths, and what is the effect?
Which factors?
Abiotic factors in these results, to the export of carbon.
Abiotic factors. Yeah. Okay. So to the first question, so this region, the production is dominated by small phytoplankton. So we did measure by classical primary productive experiment. So in parallel to this, we did the classical high volume, where you measure productivity and you measure also fractionated chlorophyll, and you see where is the productive is actually concentrated, and it is within the small phytoplankton. That was one of the reason we want to go there, because it's known to be dominated by small. So I don't know the rates of the larger cells. We could estimate it by. We could estimate it, but to answer your question, they were not the dominant ones. They were not dominating the primary productivity there in this area. For the abiotic, so some of the abiotic factors that we try to count is the time the particles are sinking, so the velocity that they are sinking, to try to implement there in our analysis. And that's very hard to do, because it's not, so we do have fluxes of the particles like carbon, particulated carbon. We try to combine that to see if that particle is largely, I see this group disappearing by pelagic, and it's because the type of particle is perhaps staying longer there, but it's very hard to account for that. So that I could not do.
More question?
My second project now. Be prepared.
Thank you, Adriana. When you compute your growth rates, you use your C14 uptake rate and then use the volume.
The biovolume by the flow cytometry.
So that's basically just for the growth rate based on autotrophy.
Yeah.
So so you don't have growth rate for the
we do. We do.
The mixotroph too?
Well, the mixotrophic is hard to do, right?
That's why I'm asking. How do you measure the growth rate of mixotroph?
We don't have. What we had is to measure by classical grazing dilution experiments, to compare with our growth rates here to see if we are not too far off, and then we were not. But I cannot tell exactly who are the mixotrophic and if they were having higher growth rates than the other one. This would be, we thought about doing by qPCI and look specifically those groups in the dilution experiments we had, but we haven't done. And I don't know now any method that could tell me that could tell me specifically for the mixotroph. But for sure that's something we would like to look, because that's the big, and also what's the cost, and if they are performing mixotrophic clearly, that's the first question that we had. Yeah. So I'm in the good track for the next project.
Adriana, I have a question. You show this model using different different, I don't remember.
Efficiency. Of the
Yeah, okay.
carbon, yeah.
And so after your results, because it means that we will not be able to do it better finally, because if it did depend on each part of the community composition of each part of the water mass, so we will not be able to fix. I am yeah, it's
Yeah. Well, we are not able to. I don't think we have that pretensions of fixing, but it is more rates, because right now there are very few rates that are considered in the models. That's a fact. And also when you consider type of cells, how much carbon they have, all this is very simplified to very few measurements. So now we have more rates available, and we can try, especially if you constrain the area. So the Southern Ocean is one area that we know there is a big gap between what is measured, what's projected. And so that's one area of concentration of some of the rates. And the Arctic as well. So we can try to constrain in some of the areas, but at the global scale, we are far from.
More question. Somebody else? Comment? So give a big thanks to Adriana. Wonderful. Wonderful. And finally, we come out to our last speaker. I didn't tell you before that this symposium has an improvement, something that is new, that we include Dr. Matias Castro and Dr. Leslie Daly as early career scientists. This is a novelty in Ecodim and in this mini symposium, because we want to increase diversity and let you know that in a couple of years you will be also being able to be here and to present and give a wonderful talk in this huge symposium. So our last speaker will be introduced Javiera from Universidad Católica, so we still Chilean by Chilean, and please.
Well, hi everyone. It's an honor to me to introduce Dr. Leslie Dayle. She's a postdoctoral research at the Genomic Ecology and Environment Center at Universidad Mayor in Santiago de Chile. She investigates the regulation of symbiotic interaction within sponge holobionts, including groundbreaking transcriptomic studies of Antarctic sponges. Leslie's journey within Ecodim highlights her remarkable growth as a scientist and mentor. She joined as a student in 2016, became a teacher assistant 2 years later, and this year it's a huge honor for the Ecodim group to welcome Leslie as a professor, a role that reflects her dedication, scientific excellence, and passion for inspiring others. Leslie's journey embodies the spirit of Ecodim, nurturing talent and building a community of exceptional scientists. Beyond the lab, Leslie's creativity shines in her love for crafting, gaming, birdwatching, and fostering scientific collaborations, traits that enrich her work and connection with the community. Please let's give her a warm welcome.
don't have my computer. I thought it was. Please, hold with me. This is for you to live the experience of field work, so you have to be patient and improvise. ¿Mm? Sí, sí, está listo. Gracias. Here we go. 3 Doritos after. Tengo un adaptador para el USB-C, servirá. Do you want one? I have some. Hey, thank you. Okay, I can do one. I remember one. So, do you know what? This is very boring. Sorry. So, do you know what does Santa Claus without the reindeers? No, but it doesn't work in English, I guess. I'm sorry. The chicken one? Okay. Why the chickens complains, you know? Because. I'm sorry. Yeah, the other one is really cool. I will tell you in Spanish. ¿Saben qué qué tiene Santa Claus sin sus renos? Insuficiencia renal. I'm sorry. They are such a good public. You should learn more. I will teach you how to laugh about bad jokes. Nope. Okay. Another computer. Oh, you got it. Aparentemente, Matías lo logró. ¿Lo voy a entrar? Okay. Let's do it. Oh, okay. Good. Yes. Vamos con eso. Sí. It's the last one. I know. That's. Thank you. Thank you for your attention. Okay. Let's try this. Tranquila. No pasa nada.
Hasta el otro lado, tengo que cambiarlo para ponerlo así, porque si no, no llega.
Yay! Thank you. Gracias.
¿No quedan enchufes?
Oh, este.
¿Has enchufado otra cosa?
Voy a usar. Y este lo dejamos.
Y lo tienes en el pendrive, ¿cierto?
Ah, sí.
Sí. Debería funcionar fantástico.
Vamos.
No lo ha reconocido, pero...
I mean, no. Yeah, I know. It's not this one, the one I put my presentation on. I mean, why work if it can fail? Thank you. So, I was just telling Marcela during the coffee break that during one of my first experiences in a course like this, one of the mantras we have, it was roll with it, because everything fails or happens during this kind of situations. And it was a great experience, so yay. Oh no. Okay. Let's just move it a little bit. Okay, good. Didn't explode. Okay, now yes. Thank you to Javiera for that kind introduction, and to the organizers to bring this opportunity to my life, and for the early careers researchers in the future. It will, for sure, that they will be glad of participating in this kind of experiences, and it's really honoring. The first time I was here was in 2014. I was starting my PhD, and I came here to see Norman Pace, and now I'm in the same scenario, so I feel really honored. And I will tell you about the story we have been building for the last 3 years in the laboratory of Nicole Treffaut. And it is about sponges and their relationship with a lot of microorganisms that in itself, it will build the alluviont. So the Antarctic marine sponges are really important in the Antarctic ecosystem because they can represent up to 80% of the demosponges can represent up to 80% of the sponges there. And they are what we will call like a bioengineer because it couples the benthic and the pelagic system, but it doesn't do it by itself. It does this job with a lot of microorganisms living in it. And altogether, this can be in association with a very generalist microorganisms, but also can be with organisms in a very low abundance and very specific taxa. And they are acquired in 2 main ways, vertically and horizontally, obtained from the environment and not from their parents. And this is where I will take advantage of Nicole King's talk of this morning because you already know how the sponges will uptake these nutrients from the water. So what you see here is a diagram of the tissue of the sponge. So through this channel, the water will flow throughout the day around 1,000 and more liters. And using the quantocytes, the sponge will uptake the microorganisms that it will be their food, using the flagella, as Nicole brilliantly showed us this morning. And after that, these microorganisms will be included into the tissue that is called mesohyl, here represented as in yellow. And they will be uptaken by the archaeocytes. And here is when happens what really got me puzzled at the beginning of the conversation with Nicole, thinking about this project is, at this point, it has to decide, and it will do it with my hands because this is the main concept that is accepted. The sponge selects the microorganisms that are going to stay as a symbiont or those that are eaten. And how the sponge distinguish between these 2 types of microorganisms is unknown. We have some clues, but we don't have a definite answer for this. And it all begins when the microorganism take contact with the sponge. This recognition and maintenance of the microorganisms are depending on the innate immune system of the sponge. And it starts with the pattern recognition receptor at the surface of the cells of the immune system of the sponge. And they will recognize the microbial or pathogen associated I don't remember the word. Membrane. Thank you, pattern. I don't know what happens. And this can go 2 ways. One, it will promote homeostasis in the case of symbionts, but in the case that it senses danger, it will generate an inflammatory response in the case of the pumps, and it will trigger this transduction cascade that would finally end up with the microbes being consumed. But what happens specifically in a sponge is these 3 types of receptors, the scavenger type system-rich receptors, the toll-like receptors, and the not-like receptors are the main ones that have been found in different sponges. Now, in the last years, we have been including in a lot of these sponges also the G protein couple receptors in this list. But it's still not really known what are specifically all those that are involved in the case of Antarctic sponges. Actually, the work I will be showing you today is only the third transcriptome that we have for Antarctic sponges. So that will tell you the little knowledge that we have in that extreme environment. But we know a little bit more about the microbes that lives in the sponges, and that is what is called a symbiotic signature in these microorganisms. And we have found mobile genetic elements, CRISPR, restriction modification, and toxin antitoxic system, and eukaryotic-like proteins. This, the mobile genetic elements is think to contribute to the establishment of symbiosis, because it will facilitate the symbiosis through the promotion of exchange, the gene exchange. But one of the main protagonists of these symbiotic signatures are definitely the eukaryotic-like proteins. Because of the similarity with the eukaryotes, they emphasize that it will be not recognized as a strange, but it is have not been identified one specific one that will allow the symbionts to actually stay in the sponge. We also have other hints about what are involved in this recognition of the symbionts, and are the ankyrin, tetratricopeptide, and leucine-rich repeats that are enriched in genomes from symbionts of the sponge. And the ankyrin repeats actually have got a lot of the spotlight because some of them have been recognized to help to differentiate between, so they will help to the immune system to be hacked by the pathogen and allow them to be surviving in this in the host. But there is one thing that is all these elements had in common, or at least the majority of them, and it is that they are all considered non-coding because they are a specific type of codification in the nucleotides ties that comprehend them that is not necessarily a product. But this is really a molecule that blows my mind since I was in the doctorate, and because of two things. First one, because they are very small, and they were considered junk when the first time we had the genome of the human being. 70% of the DNA they sequence, if you know, it was called junk DNA, and it was because none of it was coding for a protein. But there's nothing more interesting than a revenge story, right? So after several years, people was realizing that this 70% was actually regulatory and really important in the development of this organism. So part of this bacterial non-coding RNA is a housekeeping RNA, like ribosomal or transfer RNA, but also can be regulatory. And this is the most fun part. So the regulatory molecules can generate different, can be generated through the transcription of different regions of our whole genome, intergenic regions, even from a region that is actually coding, they can obtain a non-coding RNA that will actually regulate the expression of that specific transcript. So it's very complex. It can be very complex. But there are three main type of regulators in the case of bacteria. There are the small RNA, the riboswitches, and the RNA thermometers. The three of them can actually have a wide variety of reaction. Actually, all of that you can think. They can repress, they can activate transcription, translation, they can stabilize different messenger RNA and promote their transcription, or maybe have the ability to be degraded. So it's really, I will say it is crazy for me. It is really weird for me to think one molecule can regulate hundreds of proteins with the same sequence, for example. So this is one of a critical molecule, not only for the central cellular pathways in the bacteria, but also for adaptation, stress, quorum sensing, virulence. And in the case of Antarctica, this is not a bacteria, but in the case of the yeast Rhodotorula, actually the synthesis of the non-coding RNA actually helps this yeast to switch metabolism and the ability to survive in this extreme environment. But coming back to the story, so I was saying, like, these molecules can contribute to the survival of these different organisms in the host. But at this point, I want to remember, or make you remember, like, symbiosis is not always good, or not always good for everybody. So we have the three types. We have mutualism, commensalism, and parasitism. And in parasitism, it was way before, because of course we are interested in fixed solutions about things like crops. And in the case of Arabidopsis and tomato, Botrytis cinerea was one of the first ones to be analyzed in this aspect. And they discovered that a small non-coding RNA hijacked the RNA machinery, the interference machinery in the host, and it generates a suppression of the host immunity and improves the infection process in the crop. This doesn't happen only on vegetables. It also happened in mice for the case of Salmonella typhimurium, which attenuates the antimicrobial capacity and enables the maintenance of these cells inside of the host during invasion. And in the case of human, this happens for a non-coding that is transported out of the cell through an outer member vesicle, and it regulates the host immune response of the human and attenuates the response to the recognition of LPS and the OMB itself. But now you will thinking like, oh, these guys are really bad, because we like to think about these guys are bad, these guys are good, but they are not. They are both. So they can also contribute to successful colonization of the host, but in a good way. And that is the case when they discovered that the expression by Vibrio fischeri of the non-coding SSRE is involved in the development of the host and regulation of the host immune response and antimicrobial activity, with a beautiful experiment, because, well, it's Vibrio, so you can mutate this and see specifically what it will happen to the host. And what they observed, it was a misdevelopment of the squid, and they were not able to colonize the host and produce this beautiful bioluminescence that we're used to think about when we think about this squid. But not even that far for us. This was just a few months ago. A small non-coding RNA called SRCF. It was described as a regulator essential for the gut colonization, and it was regulated by our diet and by the other microbial organisms that were part of our microbiota. So it's very interesting, even for the cases, because the first one, the case of the squid, is a one-to-one interaction. But the case of humans is a lot more complex and is a lot more similar to what we observe in the sponge. So that makes us wonder if any of these mechanisms could be involved in the case of the sponge interactions with the symbionts. through the regulation of the host defense mechanisms. So we pose 3 specific objectives. The first one, of course, because we know very little about the sponge, who was characterized the functional genes, specifically those that will be involved in the recognition of these microorganisms. Second, to identify the profiles of known coding that were being produced by the bacterial community. And finally, determine if the potential host targets were actually being targeted by this non-coding RNA. But I will lead your expectations already to a lower point because I'm not going to be showing you the third part. I would love to, but it's still running. So, but in any case, these other 2 objectives were done with samples taken in the Greenwich Islands and South Shetland Islands during 2000, well, I will say that after, and in the Antarctic Peninsula. And the sponge we are going to be studying is what we identify as a Myxilla vultonancora, which is this beautiful red, bright sponge. And here we characterize the spicula that are very similar to what we have as bones, is the structure of these sponges. So the first objective, that was the characterization, we use sponge individuals from 2019, 20, and 21. We extracted RNA, messengers RNA that were a size of more than 200 nucleotides, trying to differentiate from the same cells, those that could be a small RNA from those that could be coding RNA messengers. And then after that, we enrich those that will have a poly A tail, because in that way we can enrich those that were coming from the host. We did sequencing and we've filtered those samples to try to optimize this to get certainty as we could that we were working with host transcripts. And then we did a de novo assembly, we compared to know how this assembly was, and finally we did a functional annotation of the transcripts. This finally resulted on a small section that was taken, because when we applied this classifier of reads, we found out that 21% was actually described as microbial origin. This, of course, can be subestimated because it depends on the database, but the 74%, it was the amount of reads we keep working with, around 40 millions. We did the assembly, and what we found was very interesting because it is a boring table, but it has very interesting numbers, because transcripts with, if we consider only the genes that were identified in this assembly, these are around 80,000, which is a very high number for a sponge. Even if you compare it with other animals, it is still a huge number. But this number can be, because it's a transcriptome. So if we only focus on the transcript with open reading frame, we will see that it's still a very high number, like 60 in the case of the reference, but it's around 40,000 genes in the case of the specific year assemblies. This is more than double the normal sponges will have, and this was coupled with what we find with the Busco analysis, which was a huge amount of duplication. This is definitely more than the 14 to 80 to 19% that you will normally find in sponges, even from those from all of those that were characterized, but none of them was actually from Antarctica. And reading about this kind of a strategy of duplication, we found out that this is a strategy to survive in extreme environments, like the one we will experience in Antarctica. And it was actually more dramatic in the case of one of the fish. The other authors analyzed when they compare the fish species from Antarctica and the same one from warmer waters, the duplication can be up to 100 times. So this was almost nothing. But still interesting because this is one of the hints we will not be able to expect if we only focus on what was described in other sponges. But we only could annotate up to 12% with one of the strategies. But if we only focus, without even the annotation, on what was shared between the 3 years, that taken in consideration Diego's question before, it was there were 3 years with very stable abiotic conditions, but we still found that the sheer amount of transcripts was almost identical or very close to what the amount of transcripts that were unique for each of these years. But this core of transcripts were actually represent 70 or 68% of the amount of the abundance of these transcripts when we analyze it. And even more, when we analyze it as functional annotation, these were stable when we compare cellular and metabolic functions for those that were shared and for those that were also unique. This is they have small variations, but as you can see, they are mainly the same categories. But we had, though, a very few transcripts that were very, very abundant. So we took the 100 more abundant transcript we have, and when we see this in it's a little bit disconfigured, just ignore it. So, no, you're not gonna ignore it now, there. You will see that the main one is associated with the homeostasis, which is expected, but the second one is immune system processes. So what is happening for the sponge the whole time? I told you, they are filtering seawater all the time, thousands of liters. So they have to be attent. They had to be all the time selecting or interacting with these microorganisms. So if we double click on those sections, those that were of my interest was the immune system processes, they will not tell us a lot about the recognition of these specific microbes, but they do highlights that the similarity of the transcript we found are very like those that are found in other animals and higher eukaryotes. This was reported before, that they are very similar, even though they are like our oldest living animal, like our oldest ancestor. But we also found that it's very interesting that they had a specific interspecies responses also for recognize probably the organisms, but to respond not only for the symbionts, but also for the phagocytosis of the other. And so for this recognition, we look into what the pattern recognition receptors were, and we found that those that were found in Myxilla were not as abundant in other species that were described before, but it was a great abundance of them that were related with the membrane-bound PRR domains. And when we analyze closely, we have seen that many of them are conserved, but there are differences in specific ones that are related with the G protein couple receptors that Nicole was mentioning this morning. And apparently, the more availability that they can have was also shared mostly with the low microbial abundances sponges that might suggest that they have this wider variety to make more specific recognition of microorganisms in this whole universe that could be the water once it is filtered. So the partial conclusions for this part is we have a very stable expression of the core transcriptome across the years, and suggests resilience and functional stability throughout as a species survival strategy in this extreme environment. And also, we had the identification of unique PRRs and low microbial abundance sponges, which will lead to this maybe specialized immune adaptation in sponges. So after we know what is the recognition systems in our sponge, we need to identify the non-coding RNA expressed by the symbiotic community. For that, I already told you. So we use the same sponges, pool of sponges, because it was not 3 of them, of course, but we use the same years, the same sponges. We extract the DNA for the characterization of the messengers. We took the smallest portion of the RNA to characterize an non-coding RNA. And we also took samples from the seawater that is circundating the sponge to see if any of these non-coding RNAs were enriched in the symbiotic community to make this analysis I was mentioning in the first of the slides, like, are these RNAs also a signature of symbiosis? And we did a similar strategy. We filter, we classify, but in this case, we only want to keep those that are from a bacterial origin. And then we did the assembly. We did the identification of the non-coding RNA using 3 different systems. 2 of them will recognize similarity of sequence with other non-coding RNAs. The other one will couple the sequence with the secondary structure. And the third one will only classify those that have a functional annotation. And we are only keeping those that are classified as non-coding for the 3 algorithms, and annotated them and clustering that by similarity, because we have different systems, we have different assemblies for the seawater and the sponges. So we cluster by similarity and annotate using RFAM and structure RNA using the secondary structure of their RNAs. This work was done with the help of Dr. Fernanda Manriquez, Dr. Mario Moreno, Dr. Vinicius Maracaya, and Dr. Victor Aliaga at the university. These are great contributions and great collaborators, and without their knowledge, this definitely can be done. So what we found after we extracted DNA was a very good amount of reads, but they get a little bit filter after trimming for quality, but then it gets scarier and we lost a lot of the sequences when we sort by first retiring those that were RNA from this category called like housekeeping RNAs, and also after the classification and keeping only those that were from a bacterial origin. but still, 2 millions, 3 millions, and up to 15 millions in the case of seawater was still good. So I didn't panic at all, but we only kept the 11 to 16% in the case of symbionts and 16 to 36% in the case of water. But after the assembly, we found very good numbers, a really high amount of transcripts with the size that we were expecting according to the methods. And when we identify this non-coding RNA, we found that for sponges, we using these three methods I mentioned before, we found that the majority of them for the associated microbes were checking these 3 boxes. But in the case of seawater, it was actually one of the lowest. And I have an hypothesis for this. Probably it's just because we know a lot more about those that are in the water. Those that were symbionts, I mean, we enrich our database with what we have at the lab, but it improved, but it still is very limited. But anyway, even though we had that fact in consideration, we have that this gold standard, it was met by the sponges associated microorganisms for 36,000 non-coding RNA and for 22,000 in the case of seawater, showing an enrichment of non-coding RNA in the bacterial symbiont community. So this will actually be considered as a signature, as we were mentioned before. And now I try to compare them, of course. How many of them were actually shared by the bacterial community from the water and those that were from the sponges? And this happened. So they are very, very unique profiles. They share very little amount of this non-coding if we consider this clustering at 80% of similarity. A lot of them were, so the total amount of them was 36 in the case of the sponge alluvium and 22 in the case of the sponge. So 7,000 of the non-coding RNA were actually redundant. So it will be classified in any of these 3 categories. But those that were unique and those that were from the seawater were still annotated. And we found 2 things. First, we found different types of mechanisms associated with the profiles. We will have, of course, some of the code-keeping non-coding RNAs that I was mentioning before. None of them, not all of them were filtered, of course, because it will depend on the availability of the data. But we found the CRISPRs that we were talking before. We found different of the more associated with eukaryotes non-coding RNAs, which are probably coming from the host, or like I'd like to think, hypothesizing like this, it might be something that will be acquired by the bacteria from the host as the eukaryote-like proteins. In this case, it will be like non-coding eukaryotes-like proteins. But my first interest, of course, it was those that I know are small non-coding, because those I know are characterized for bacterial communities. So there was still an enrichment in this case for those that were coming from the symbionts, but we are working from there for those that are actually more than 80% of the coverage of the transcript annotated as a non-coding. We'll still have around 600 of non-coding for the next step, which is a lot. if we consider that the amount of transcripts related to the immune recognition is around 10,000 of genes, so we have to do one-to-one comparison in a way that is first analyzed by the similarity, and secondly, by the secondary structure that can actually able or enable the transcription or translation of those proteins. So the partial conclusions for this point is that we have an enrichment of non-coding RNAs, and both of the candidates being unique to both symbionts or the surrounding seawater, revealing additionally a remarkable diversity of non-coding RNAs in both systems. So in my case, it is not a full story because I'm an early career researcher, as a lot of you are. So I made this for you to don't feel bad about probably the state of your research. So the perspective for this is keep working on the potential taxa from for the origin of this non-coding that are found in the profiles and how their abundance, but mostly to identify where are the potential targets in the host using the immune-related transcripts. So that's the story I wanted to tell today. And this story will not be able to be done without a lot of help, a lot of people involved, and a lot of fundings. And that's it. Thank you.
Thank you very much. Thank you very much, Leslie, for your wonderful presentation. I am sure we have a lot of questions and that there are many things to be answered.
Okay, thank you. So, what would you like to, or what are you expecting to find, or what would be the discovery that you think would be, oh, that would be fantastic if I find that? Maybe you won't, but what are you expecting for your third point? What will be your ideal situation or ideal discovery?
My ideal discovery, I would definitely like this to be a yes for this hypothesis, because I mean, it is okay that the sponge lead this selection. It's okay. Everybody can decide what they want to eat. But I like to give the power to bacteria, so I will be very happy if we found some of the non-coding RNA to interact and have a target like HFQ or proteins that will facilitate the establishment of the microorganisms inside vacuoles, for example, and that will allow them to avoid phagocytosis and then to stay there, do their job, and stay there like paying their rent, but not necessarily because they were chosen. It's because they decide to stay as a way to both can survive and a way that have been growing this interaction over time. I mean, we have sponges for a long, long time here, so it is not weird to me to think about this story I was telling you before. Like, this maybe started as a pathogenic interaction, but after many, many interactions, finally it was, I don't remember who mentioned, I think it was Matthias, like viruses, it will take a little bit of something from their host when they do the lysogenic cycle. So it will not be weird for me to think about pathogenic becoming something that is recognized as something different than can be hijacking their way in. So that will be my...
Are you planning to do some experiments? Control experiments?
I would love to. But I mean, the project is ending, but I would love to keep this going. So I think there are a lot of possibilities with synthetic biology right now. We don't have culture of cells for the sponges. We have a prototype that have been published by other researchers for primary cells culture that can be used as a model to test this and do some control experiments. But it is a matter of time, fundings, and things like that. I mean, it will be great.
Thank you very much. More questions?
Thank you, Leslie, for your presentation. My question is also, I think, is very basic because this is completely out of my expertise. How do you think is this play would happen in the different phase of the growth of the spongy? Because I guess also the community is acquired depending on the if it's a juvenile adult. I mean, so how do you think this would play in the different phases?
So I think if this is the case and a non-coding will be involved in the acquisition of the symbionts, I think it will be working, as I was telling Osvaldo before, as an evolution of different interactions, but these interactions are local, and so it will be the same, but for different communities. And what we have reported, different scientists have reported for the sponges, are they are selecting different specific communities in each of the local cases. And in the specific case of one species, if you would compare it, it would probably be the same, because if they are recognizing, if they had the pattern recognition receptors for specific microorganisms, they will be recognizing, always interacting with the same kind of thing. And that will lead to all this process. But it's completely unknown, yeah.
I'm like Adriana, it's really way beyond my expertise. But one question, and actually it's a specific question for your research, but also more general question, is that is there anything specific with Antarctic sponge, or the mechanism that what you are investigating is not different in tropical sponge or temperate sponge or deep sea sponge versus more, so is there something really specific about Antarctic sponge, or I mean, I understand very well that they are not known, so that's quite
So the quick question is, I don't know, but the longer answer will be that it might be specific for Antarctic because they are exposed to a more extreme pressure, and maybe they had during time to improve their ability to infect and survive in this environment as bacteria, or not having all the capabilities to deal with the environment by itself. And this strategy to live associated with another organism will be better, but I don't, sorry. I would love to have an answer.
But the mechanisms that you are studying have been studied also, these non-coding RNAs have been studied also in non-Antarctic sponge, or it's no, okay.
No, in sponges, not at all. They have been studied in different cultures, like crops, humans a lot, because a lot of interest is in how the gut is established and how they influence the immune system of humans. And specifically is very well known for pathogens like Salmonella typhi and Typhimurium, Pseudomonas aeruginosa, but definitely not for sponges. I know there are some people making progress on the eukaryotic-like proteins was very interesting. This Laura Schindler from Israel was presenting in ISME last year about this protein that was a eukaryotic-like protein that actually is associated with the Golgi system and the export and formation of outer member vesicles, which I was like, this is amazing, because this kind of thing is not necessarily one only answer. It can be that eukaryotic-like proteins or all these signature marks that we have for symbionts are working together. So eukaryotic-like proteins can favor the production of the outer membrane vesicles that can actually transport this non-coding RNA to the host to hijack all the systems. But again, I don't know.
More question from the student? Something that you don't understand and you want an explanation? What are the sponges? What is symbiosis?
Do you know about the sponge?
Hi, Leslie. Thank you
Hi.
for your presentation. I was curious about this graph that you showed between the sponges and the seawater, and they had in common like 77 only.
This one?
Yeah. Could you like enter more
Yeah,
in
yeah,
the specifics,
of course.
because why so little? Because you said that we know a lot more about seawater than in the sponge.
Yeah.
But
So for that comment specifically, it was about this. So all what we sequence afterwards, we pass it through these algorithms that will tell us, okay, is this an encoding? Is this an encoding for the positive or the negative strain? And we will be classifying in each case which are the criteria that are meeting. And for the case of sponges, we have that the majority of them were actually meeting the three, like our gold standard. But for the seawater, we actually lose the majority of them. If you, maybe you, I don't know, it's very small, but it says blast X. So that was one method I mentioned that will classify them as coding or non, or like coding for a protein, not analyzing the structure of a non-coding RNA, but telling us this was described before as a coding protein or transcript. So if you see, the main amount of transcript in that case were lost because they only met the two other algorithms and they were not classified as non-coding by BLAST-X. So that is the reason I said, like, we know a lot more about what is in the water. So we have a lot more of those transcripts described as coding or in our databases. But even though we still have quite an amount of non-coding for the seawater, 22,000, and for the sponge all around 36,000. So from all those, because they were from two different assemblies, the transcript have different names. So we cannot filter just saying, oh, one and one are shared or not. So we have to compare the sequences of these ones. So we use a clustering system and we apply a 80% of cutoff. And those that were common and were classified as the same cluster, we say, okay, this one is 1, this one is 2, and this one is 3. And after we perform the band analysis, and that's what I said, like we lost 13% of this non-coding total because they were the same sequences. And from those that are not classified as unique non-coding RNA, I mean,
unique. We have that 29% were unique, only reads from the seawater were found. Only 29%, 29,000 were found, and those that derived from bacteria from the oleoviant, and only 77 were found to be shared between the two profiles. That does clarify.
Thank you. I think we have time for a last question.
Yeah, no, while you were explaining it, it occurred to me that so we use a lot of the ITS, right, in our studies to and it's also non-coding sequence, and for what we do to look if the cells are able to reproduce, so if they are isolated or not, so then describe species, etc. We use the secondary structure. That's the most important for the ITS, not actually the sequence. So I'm wondering if there are also studies or databases that show different secondary structures of the non-coding RNA and how that's related to their function.
Yes, that is actually the way we identify these ones. So we use what is called Inferno, what is for RNA, and this will consider not only the sequence, but also the secondary structure and how are their function, and if they are only a sequence or they have active ribose switch activity, or if they are a small RNA, or maybe a housekeeping non-coding. Thank you.
Thank you very much. So, thank you, Leslie.
Thank you.
Wonderful talk, wonderful talk. Now we are ready for all the presentations. I want to say thanks to all our speakers. Now we have a small coffee and come back for a final discussion, and I hope you have written all your questions or your comments or the things that you want to continue to talk with the speakers. Thank you very much.